All righty, let's go ahead and get started. Any questions before we get rolling? All right, as you know, things are winding down rapidly. We've got the entire rest of our schedule here on the board. So again, today, we do have a little lecture we need to finish off. And the good news is most of it is going to be reviewing information we kind of already know and synthesizing that information. Because our primary focus for today is to catch up on our brain anatomy that we haven't had a chance to get into yet. I don't think we'll get to the spinal cord today, but if we do, that's just bonus. A handful of assignments still left to do. Your Unit 13 review is due on Thursday when we come back from the day off tomorrow. I am hoping to get to our cranial nerves on Thursday. If not, definitely Friday. I want to make sure we get that done before the weekend, because as I mentioned, that's going to easily be about 25% of your lab exam. Also on Friday, your PhysioX exercise 3 is due, which has 9 activities. So hopefully you've already started on that. If not, you'll be busy tomorrow. Monday, next week, we still have your 15-point nervous system review. Again, it's exploring that relationship between structure, function, and location. So that one is going to be graded for correctness. So make sure you're looking at that. And then Tuesday, your last assignments, Unit 14 review is due, and then also that reflex lab is going to be due as well. So again, you're using your lab manual, doing the procedures that are in there. Again, any procedure that you're not capable of doing, either because you don't have equipment or don't have a willing participant or however else that it works out, do it as a thought experiment. Explain what you think would happen when you do those things. So I want it all completed. I want it filled out, and that is due on Tuesday. And then the rest of it's exams. On Thursday, you have your lab and lecture exam. You've already seen the format of those. You've taken 3 of those, so you're just going to keep doing what you're doing on that. And then on Friday is our final exam. As I mentioned yesterday, it is 100 multiple choice questions. You will have 100 minutes to complete it. You will have the entire class time to complete it. So anytime during 8 and 12:35, you can start that. Just make sure you give yourself enough time to complete it. All right. And then on August 1st, you get to rest. All right. Questions on any of that? All right. Let's dive in where we left off. So, as we talked about, I need that. We have so far discussed how out here on the cell body and the dendrites, you would receive a signal, some type of stimulus. That stimulus would open chemically gated channels or mechanically gated channels. Say it this way. And you would use that to produce a graded potential. And then that graded potential is going to spread to the axon hillock. We have talked about how, and I'm going to cheat in my drawing of this. At the axon hillock, we discussed how that stimulus of the graded potential can be used to produce an action potential. Right, that big positive all or nothing signal. We then talked about how we can propagate that action potential down the length of the axon. And we talked about being able to do that 2 ways, both with myelin sheath and without myelin sheath. We talked about the differences between those two. So really the only thing we haven't really talked about yet, and in fact we actually have talked about, is how this neuron is going to communicate with its target cell. So again, the whole point of producing this signal, generating this action potential, spreading this action potential down, is so that our neuron can communicate with its target cell. And just out of curiosity, what do you think the most common target cell is? What do most neurons communicate with? Oh, wait. Well, what is the most common functional type of neuron? How many different functional types of neurons are there? Let's start super easy. How many different functional types of neurons are there? Okay, there's two, sensory, interneuron. What's the third? What is the third functional type of neuron? Sensory, interneuron, and what's the third? Come on, people. Motor, there you go, excellent. So there are three types. Of those three types, what is the most common? Sensory, interneuron, or motor? Interneuron, excellent. So what was the function of interneurons again? Excellent. Transmit impulses between what? Between the sensory and the motor, right? Yeah, they connect the two. They connect the motor to the sensory, right? So they're the sensory to the motor. They connect those two together. So that means if they connect neurons together, if interneurons connect neurons together, what is the most common target of an interneuron? What do most interneurons talk to? Yeah, they pass information to what? Other neurons, exactly, to the within the brain. So within the brain, so the most common target is actually another neuron. So most neurons talk to another neuron. Of course, we need a name for that special location, specialized structure, where a neuron communicates with its target cell. And what is the name we gave that specialized structure where a neuron communicates with another cell? Synapse, exactly. So that's what we need to talk about next, or really what we need to talk about last. We have produced graded potentials, we've produced action potentials, we've propagated that action potentials. Now we need to communicate. The last thing we need to talk about is we need to talk about how you communicate with your target cell. And the good news is you've already learned the two ways that we are going to do this. So really, there's not a lot of new information right here. We're just synthesizing information that we have already learned. So let's do that. As was mentioned, our last goal is to communicate with our target cells, transmit that signal from one cell to another. And we do that at a synapse. The synapse is the functional junction between the presynaptic neuron and its target cell. However, as we already mentioned, the most common target cell is another neuron. But it doesn't matter what it is. It could be a gland, it could be a neuron, it could be a smooth muscle, skeletal muscle, cardiac muscle, whatever it is going to communicate with a synapse. And even though we have not used the term before, we have actually talked about the fact that there are actually two different types of synapses. The first is an electrical synapse. Now, as I mentioned, we have not used the term electrical synapse before, but it is a concept we understand because, as we've talked about, two cells can be held together by connexons. So you have a connexon in one cell and a connexon in the other cell and another connexon here and another connexon here. And you put these connexons together and we get a fluid-filled pore between the two cells, allowing the cells to rapidly communicate with each other, carrying information back and forth between those gap junctions. And we talked about there being some advantages to that. In this case, the cells are basically hardwired together. So they work together as one. Whatever happens to one cell happens to the other cell. Another huge advantage of this is it allows for two-way communication. So for instance, if something wonky happens in one cell, it can spread to the other. But at the same token, if something happens in the other cell, it can spread back to the first. So it does allow for two-way communication with that. And so let's go ahead and move those into our gap junction to emphasize that. Another huge advantage of this is it is a fast process. Right. Spreading ions or other things through this fluid-filled pore is a very fast process. So there are some huge advantages to these gap junctions, as we talked about when we first discussed them. We didn't use the term electrical synapse when we talked about it, but gap junctions are definitely something that we have talked about. However, there is a downside. Remember when we talked about gap junctions, one of the places where we didn't say there were a lot of them is in our nervous tissue. We talked about it being in smooth muscle. We talked about it being in cardiac muscle. We talked about it being places in that, but we said there weren't a lot of them in the nervous system. And part of it comes back to that concept of them being hardwired. There's an advantage to things being hardwired in that they work together as one. But the downside of being hardwired is that it's not modifiable. You can't modify or make decisions. There's no decision making that takes place. Your house is hardwired. When you flip that light switch, your light comes on. There's no decision being made. The light doesn't decide, oh, maybe when that flip switch goes on, I don't want to turn on today. No, it's got to turn on. It is hardwired. There's no decision making that can take place. And one of the vital things of our nervous tissue is that we be able to make decisions. Because as we used the example before, if I stand here with my mouse in my hand over Zoom and tell you I am going to kill you, and a bear with an axe walks into your room, looks you in the eye and says, I am going to kill you, you are going to respond very differently to that stimulus. You're getting the same stimulus twice, but you are going to respond very, very differently to those two situations. So the advantage of gap junction is speed and coordination. The disadvantage is that we can't modify it. We can't make decisions. So as we've talked about, most of our nervous system instead takes advantage of, so we did all of that, chemical synapses. A chemical synapse is something that we have talked about already. We saw the example of it in the muscular system, But we can also simplify our illustration of this as well. We know a synapse is going to have a synaptic end bulb. Right, this is our presynaptic neuron. Oops. Excellent. And what were some of the specializations we know that occur in our synaptic end bulb? There are two main specializations. What were the specializations? I'm not going to bother drawing them. I'll just write them. Excellent. We have voltage gated calcium channels. That is definitely one of the specializations of our synaptic end bulb. And what was the other specialization? Vesicles containing neurotransmitters. Excellent. Perfect. Excellent. We know the neuron does not touch the target cell. So let's go ahead and draw a target cell and make it purple just for the heck of it. Excellent. So here we have our target cell, and we know these two don't touch. There's a space in between. And what do we call the space in between? Synaptic cleft. Excellent. So we have that synaptic cleft in between the two of them. And then our target cell also needs some kind of specialization. What is the specialization that our target cell is going to need? If our synaptic end bulb is going to release a chemical signal, what does our target cell need? Chemically gated channels, absolutely. Now, technically, they don't have to be cation channels, right? Because again, different cells, we want different effects, but you absolutely have the right idea. What we are going to need is some chemically gated channels. So that what happens is that neurotransmitter binds to them, allows ions to flow. So where do I want to write this? I'll write it in here. Our neurotransmitter, I'm going to abbreviate, binds to receptors. When it binds to the receptor, it opens channels, ions flow, and they either in or out of the cell. And of course, as we know, that produces a current. Oops, no, wait, what's going on up there? All right, binds to receptor, opens channels, ions flow in and out. There you go, that produces a current, which we call a potential, a graded potential. Or we can also give it another fancy word. We can call it a postsynaptic potential because it occurs in the postsynaptic cell. This is our postsynaptic cell. All right, so notice when we talk about our chemical synapses, it has a much more elaborate anatomy. So it is much more elaborate. They don't actually touch. This isn't hardwired together. Because of that, there's a couple implications to this. The first is that it is going to be slower in the spread of the signal. Instead of the two cells just sharing sodium or something like that, a whole lot more has to happen. I don't know how many of you actually went and found one of those reaction time websites yesterday. But if you did that, as I mentioned, part of that reaction time that you see is the spread of the signal down the axons. The other delay in that reaction time is the processing that takes place at a synapse. So the amount of time it takes you to do an activity, right, that instantaneous, it isn't instantaneous, there's a little bit of a delay, is due to the propagation of the action potential and the time that it takes to communicate at that synapse. Also, notice this way, we have a presynaptic and postsynaptic cell. There is only one-way transmission of information. This information in this case only goes in one direction. It only goes from the presynaptic cell to the postsynaptic cell. So it's slower. It doesn't coordinate the cells the same way. It doesn't allow them to talk back and forth between each other. But like I said, the huge advantage of this is that this signal can be modified. That allows us to change our behavior. That allows us to make decisions. And so while maybe a little slower, maybe a little bit more complicated, it is much more powerful in what it is able to do. All right. So again, cells don't touch. The presynaptic cell releases a neurotransmitter. There's a delay in this process. It only transmits one way, but it's modifiable. It can produce different types of reactions. We can make decisions in producing that graded potential in the postsynaptic cell, or again, what we called a postsynaptic potential. And here's the best part. The best part is you already know how it works. Notice here we have a neuron communicating with another neuron instead of a skeletal muscle cell. But the exact same process we talked about for communication and communicating at the neuromuscular junction is exactly what's going to happen here. An action potential spreads down the axon, depolarizes the synaptic end bulb, opens voltage-gated calcium channels, calcium rushes into the synaptic end bulb, calcium triggers the vesicles of the synaptic end bulb to bind with the plasma membrane and release their neurotransmitter by exocytosis. That neurotransmitter diffuses across the synaptic cleft, binds to receptors on the target cell, opens those receptors, and ions can flow into or out of the target cell. We don't have to bother writing out this process and learning this process again because you already know it. So that process you had to learn for the last part of the exam, for the last exam. And again, because of the variety of questions you guys had, some of you actually had to describe this process on the last exam. Well, guess what? You may have to describe this process again on this exam because this signal transmission at a chemical synapse is exactly what we saw in the last part. So we learned it once, we get to use it twice. All right, questions on that?
Yeah, I have a question.
Yes.
I was a little bit confused on the homework on one of the questions where it's saying that the action potential is generated at and then it gives the options. One of the options was the trigger zone of the exon, and then the other one was the exon terminal. And I also thought that it was being created at the exon hillocks. So,
well, remember the trigger. So the trigger zone is within the axon hillock. So the axon hillock is the name here. Let's just we'll draw it. It'll be simpler. The axon hillock is the portion of the, or let's say it this way, the transition region, the structural transition region where the cell body changes to become the neuron. So again, the axon. So it's this part right here. Part of that transition is simply structural, where we need the neurofilaments to form it. It's narrowing, it's becoming more hollow, it's not branching the same way that the cell body is. But the other big specialization is the transition of the plasma membrane. Remember, the plasma membrane of our axon has a massive number of voltage-gated sodium channels and voltage-gated potassium channels, and there are none of those on the cell body in the dendrite. So the axon hillock is this transition region where it goes from having no voltage-gated sodium channels to the part where it's going to have some, right? Most, all, massive numbers of them. So it is a bit of a transition region. So it is structurally a transition region. Now, just adding one voltage-gated sodium channel to the plasma membrane isn't going to allow you to produce an action potential. Neither is adding two. Probably not adding five. So within this axon hillock, as we're getting more and more voltage-gated channels on them, there happens to be a functional area. Again, it's a functional area. It isn't something we can see. I guess technically, if we could get all the way down to the molecular component where we could actually see the receptors on there, we would be able to see the area that where they had become dense enough where we're like, all right, now they're dense enough where we can produce an action potential. But we're not able to see at that level. So we know functionally within the axon hillock is a region we call the trigger zone. So that functional area in the axon hillock is the trigger zone, and that trigger zone is where the first action potential will be produced. So the trigger zone is within the axon hillock.
Okay. Okay. So the action potential is generated in the trigger zone of the axon, correct?
Correct.
Okay. Gotcha.
Yeah. Now you're right. Wording would have been better if they said the trigger zone of the axon hillock. That would have been, you know, a more appropriate wording. But technically, the trigger zone is a part of the axon as well, because the axon hillock is that transition region.
Okay.
Okay. All right. Any other questions on this? All right, excellent. So what we can do then is focus on that postsynaptic potential. Again, binding of the neurotransmitter to the postsynaptic cell opens up ion channels. Ions either flow in or out, producing a current. And as we mentioned, that current, I'm not sure why that's so small, we can call a graded potential. Now, or a postsynaptic potential. As we've talked about, the goal of this, since if you think about it, the goal of a graded potential on a neuron is to help it decide if it fires an action potential or not. Right? This is where we make our decisions, or how we make a decision. Do we fire an action potential or do we not fire an action potential? That is the goal of these postsynaptic potentials. So not surprisingly, oops, and I have to move that. Not surprisingly then, when we talk about these postsynaptic potentials, they can be depolarizing or excitatory, or hyperpolarizing and inhibitory. Now again, fun with definitions. Depolarizing, of course, means it makes it more positive, right? We've done that, we've explained that. In this case, excitatory means that it makes it more likely to fire an action potential. Hyperpolarizing, of course, as we have defined, hyperpolarizes, let's be consistent, means to make the cell more negative, and inhibitory means that it makes the cell less likely to fire an action potential. So obviously they kind of mean the same things, but technically the definitions of these two terms are slightly different. Let's take a look at a pretty picture. Here we see an example of an excitatory postsynaptic potential. Again, it causes the cell to depolarize. The cell gets more positive. And notice the key to our definition here. It makes the cell more likely to reach threshold, more likely to fire an action potential. Did I say it has to get it to fire an action potential? No. To be excitatory, it does not actually have to get it there. It just has to make an action potential more likely. So notice the example of the stimulus here. The stimulus here doesn't actually get to threshold, but even though it doesn't get to threshold, it would still be considered an excitatory postsynaptic potential. And remind me again, what types of channels? Are we likely to use to produce an excitatory postsynaptic potential? What ion are we going to open? What type of ion channel are we going to open to make the cell more positive on the inside? Sodium, exactly. So typically uses sodium channels. There we go, and perfect. So typically this involves sodium channels. Conversely, an inhibitory postsynaptic potential, right, makes the cell more negative, causes a hyperpolarization. Again, it makes the cell less likely to reach threshold, but it could still potentially, from other stimuli, be able to reach it, and this would still be considered inhibitory. So this one just moves it further away from threshold, making it harder for it to reach threshold, and therefore it is considered inhibitory. And what type of ion channels did we say we would most likely use to inhibit that postsynaptic cell? Type of ion channels would we want to use here? Potassium, anything else? Chloride, excellent. So these are typically going to involve either chloride channels or potassium channels. All right, now, the reason we say more likely and less likely to reach threshold is because typically one graded potential is not enough for the cell to reach threshold and generate an action potential, right? Why is that? Remind me again, how much of a voltage change do we typically need to reach threshold? Well, remember, 100 millivolts is the size of the action potential. What do we actually need to reach threshold? 10 millivolt change, a positive 10 millivolt change. And what did we say the average graded potential size was? Anyone remember how big, what the amplitude of a single graded potential? Yeah, right, 2, plus or minus 2, right? So we said about there. So even if just simple math, it sounds like you would need at least 5 excitatory postsynaptic potentials added together to reach threshold. But that's where the decision making comes in. A single neuron typically has somewhere between 1,000 and 10,000 synapses on it. So a single neuron at any one time could be receiving 10,000 pieces of information. And really, in what a neuron is it's a bean counter. It's an accountant, right? If you have 9,000 positive signals coming in and 1,000 negative signals coming in, are you going to want to fire an action potential? 9,000 excitatory signals, only 1,000 inhibitory signals. Are you likely to fire an action potential? Yeah. If you have 8,000 negative signals coming in and 2,000 positive signals coming in, are you going to fire an action potential? No. And what happens if you have 5,000 excitatory and 5,000 inhibitory? Are you going to fire an action potential then? No, probably not, right? Again, its job is to count beans. And essentially, one excitatory postsynaptic potential is canceled by one inhibitory. So if you think about it, 5,000 positive, 5,000 negative, they cancel each other out. That's what your accountant does, right? You make $5,000 this month, you spend $5,000 on Pokemon cards, right? Your net for the month is $0, all right? So that's what they do. They count beans. They add these positive and negatives together and see if we get a signal big enough to reach threshold and fire an action potential. This is how we process information. This is how we make decisions. We collect thousands of pieces of information, some positive, some negative, and we do the math. If there's more positive and the more positive is strong enough of a signal, we fire an action potential. If it's not, we don't. And that's basically what a neuron does. All right. Now, your book's got a nice analogy for how this works, this adding together of signals. I'm going to cheat and steal some of the screen here so that I can, so I'm going to go ahead and just do that. We have to reach threshold. And the example your textbook uses is to have a bathtub. You have this bathtub and threshold is filling the bathtub. Your goal is to fill the bathtub up. All right. The problem is that you don't have a plug for the drain. So let's say, for instance, it takes 10 buckets of water to fill up the bathtub. After all, it takes 10 millivolts of change to reach threshold. It takes 10 buckets of water to fill the bathtub. If my faucet is in the basement and I go and I fill my bucket in the basement, I bring it up to the second floor of my house, I dump it in the bathtub, and then I run back down to the basement, fill the bucket, and come back up to the second floor. If there's no plug for the drain, is there going to be any water left in that tub when I get back? No. So if I try to fill it that way, I'm never going to be able to fill it. So what I need to do is I need to get buckets of water into this tub quickly. And there's basically two ways that I can do that. The first is me working by myself. Me working by myself, if I don't have the faucet next in the basement, but if I have it right next to it, I can fill a bucket and dump, and 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 try to do that as fast as I possibly can. Right? To go even faster if I have two buckets. While one bucket's filling, I could be dumping the next. And then I can grab that one and fill and dump the next. So if I can do it by myself quick enough, if I can have one person, or in this case, one neuron, stimulate the target cell fast enough, we can reach threshold. I can fill that tub and I can reach threshold. But do I really have to do it by myself? Maybe I want to take a bath. And so what I do is I call up 10 of my closest friends and invite 10 of my closest friends over. And me and all 10 of my closest friends all grab buckets, all fill those buckets, and we all dump those buckets together at the same time. If I do that, can I fill up the tub that way? Yeah, absolutely. So instead of just relying on one neuron, I can have many neurons all firing together. And when all those neurons stimulate it together, we can get a big enough signal. Let's see, we can stimulate together. And we can get a big enough signal to reach threshold. And so those are the two ways that we can do this. Of course, we have fancy names for these two ways. Temporal summation, temporal referring to time, is one neuron stimulating the target cell many times very rapidly. So the single neuron stimulates the target neuron 6, 7, 8, 9, 10, 12, 14, 15, 20 times in a row, really quick in time. So it's fast in time, and when it does it fast in time, we're able to produce a big enough signal where it reaches threshold and it produces an action potential. Or I can grab my 10 friends and we can all be together in the same space. And if I grab all 10 of my friends and we're all together in the same space, we have multiple neurons all stimulating at the same location. And when they all stimulate in the same location, they produce a big enough signal to generate an action potential. So basically there's two ways to count beans. Count them in time temporally and count them in location spatially. Questions on that? All right, excellent. The last little bit we need to talk about for lecture today, and then we'll move on to our lab, is to talk about neurotransmitters. As I mentioned, there have been over 100 different neurotransmitters that have been identified in the human body. As we've also talked about, their effect of the neurotransmitters doesn't depend on the chemical itself. It actually depends on the channel. So let's see a couple quick examples. Excellent. We know, oh, here, let's finish the picture. We know that our somatic motor neuron communicates with our skeletal muscle cell. And remind me again what neurotransmitter is released here. What neurotransmitter is released from our somatic motor neuron to communicate with our skeletal muscle cell? Acetylcholine. Excellent. We know it binds to a chemically gated cation channel. And when it binds to that chemically gated cation channel, does it have an excitatory or an inhibitory effect? Excitatory. Right? It stimulates the skeletal muscle cell to fire an action potential. It depolarizes the cell and excites the cell. Cool. So that means acetylcholine definitely is excitatory. Except something really, really interesting happens. If we take that exact same acetylcholine, instead sprinkle it on a branched looking cell that happens to also have stripes on it, what type of tissue might this be? Cardiac muscle, excellent. If you were to take that same acetylcholine and you were to sprinkle it onto cardiac muscle, then what happens with our cardiac muscle is that it actually slows the heart rate down. Acetylcholine is actually inhibitory on cardiac muscle. So how could it possibly be excitatory in one place and inhibitory in the other? It's the exact same chemical. So how is it different? What's different about these two situations? Different channels, exactly. As it turns out, this acetylcholine channel in cardiac muscle is a chloride channel. It's a chemically gated chloride channel. So when our acetylcholine binds to it, it opens up chloride channels. Chloride rushes into the cardiac muscle cell, and the cardiac muscle cell is inhibited as an effect, and your heart beats slower. In fact, that is how our parasympathetic motor neuron slows our heart rate down. It releases acetylcholine and slows down the heart because it opens a different type of channel. So when we talk about neurotransmitters, we talk about them being inhibitory or excitatory, but really it's the channels that determine that. Different channels open, different channels have different effects. So here, the exact same chemical, acetylcholine, in one instance is excitatory, in the other instance is inhibitory. All right, questions on that? All right, so let's talk about some different types of neurotransmitters. Oh, and of course, talk about different ways the effects of the neurotransmitter can be modified. I guess I need to, well, we'll redraw it this way. Target cell, axon terminal, vesicle. Excellent. All right. One of the important things to understand is that the relationship between a neuron and its target cell is dynamic. It's changing. And there are a couple ways we can get a stronger or a weaker effect. One way is obviously to produce more action potentials. If you have more action potentials in the target cell, you are going to release more neurotransmitter. And if you release more neurotransmitter, you obviously get a bigger effect. That's an obvious way that we can see some difference. Here, and let's actually do this. More action potentials. So I can spread that out that way. And I can erase that. Perfect. Oops. All right. So obviously that's one way I can affect the relationship between these two. But there are other ways I could do it as well. One is I could make more neurotransmitter. Right? If I filled the vesicles with more neurotransmitter, then obviously I'm releasing more neurotransmitter. And if I release more neurotransmitter, I get a bigger effect. And of course, the opposites of these are true as well. If I send fewer action potentials, I get a smaller effect. If I make fewer neurotransmitter, I can get a smaller effect. I can also make more vesicles. If more vesicles are released, if more vesicles release their neurotransmitters, right, then obviously we release more neurotransmitter. And of course, get a bigger effect. So All of these are ways, notice, from the synaptic side that we can, from the presynaptic side, that we can make a stronger effect. But we can also affect the strength from the postsynaptic side as well. And the primary way we do that is by modifying the number of receptors on the plasma membrane. Notice if our postsynaptic cell just has one receptor on it. If I dump a whole bucket of neurotransmitter onto that cell and it only has one receptor, is that going to have a huge effect on that cell? If I can only open up one channel and only let one channel's worth of ions flow in, is that going to give me a big effect? No. But if I have a million receptors on the surface, if I sprinkle even the tiniest bit of neurotransmitter on it, is it likely to bind to one of those millions of receptors? And am I likely to get an effect from even a tiny amount of neurotransmitter? Yeah, absolutely. And the awesome thing about it is dynamic as well, right? These are constantly changing, right? When you have that brand new baby, right? If they just whimper even the tiniest bit in the crib, right? You're instantly aware, instantly paying attention, and give them your undivided attention, right? Once they get to be teenagers, they could be standing next to you, yammering in your ear for a half an hour, and you're hardly aware they're there, right? We have this ability to habituate. And what happens in those cases is that we drop the number of receptors. As we drop the number of receptors, we become less sensitive, right? Think of it in the extreme case. If I get rid of all of the receptors, now I'm completely insensitive to that neurotransmitter, and that neurotransmitter no longer can affect that cell. So how many receptors we have on the plasma membrane is very dynamic. And we can change it, therefore changing the relationship, changing the strength of the signal that we get from that presynaptic cell. So modifying a synapse, modifying that communication happens both on the presynaptic and on the postsynaptic side. All right, so again, we can change the number of action potentials, how much neurotransmitter we're making, how many vesicles we release. Oh, wow, we can even affect the synaptic cleft, right? If we move the cells further apart, it diffuses off more rapidly. Closer together, they're going to stay there, and it's more concentrated. One of the essay questions I know some of you got was, what happens if you removed acetylcholinesterase from the synaptic cleft, right? If you get rid of that enzyme that's in there, then the neurotransmitter stays in the synaptic cleft for a longer period of time, giving you a bigger effect. And then, like I said, also the number of postsynaptic receptors. So there's lots of ways we can change the strength of the communication between this neuron and its target cell. And then, of course, we've also talked about toxins like curare, like tetanus, curare and tetanus are the two big ones that actually bind to the receptors and either bind and block or bind and activate. All right, so like I said, over 100 different types. I'm not going to obviously make you recognize and identify all 100 types, but I do want to talk about some of the more common ones. We've already talked about acetylcholine, and acetylcholine is the most common neurotransmitter in the peripheral nervous system. And notice, as we also hinted at, acetylcholine can be either excitatory or inhibitory, depending on the receptor. But notice I said it's the most common in the peripheral nervous system. That means it's not the most common in the central nervous system. Those fall to glutamate and gamma aminobutyric acid. Now, one of the things that I've emphasized in this class is spelling matters, and you're not allowed to abbreviate things, but this will be one of the exceptions I will allow you. GABA, G-A-B-A, is an appropriate name for gamma. I mean, I'm not going to make you memorize gamma aminobutyric acid. you can just use GABA. So just know that GABA is the most common inhibitory and glutamate is the most common excitatory in the central nervous system. There we go. Some hormones are larger than just small molecules, things like epinephrine and norepinephrine. Epinephrine sounds familiar. Is there another name for epinephrine?
Adrenaline.
Adrenaline, absolutely. So adrenaline and noradrenaline, or epinephrine and norepinephrine, right? Those are the hormones that are involved in what process? What is epinephrine or adrenaline involved in? When do you get an adrenaline rush? Yeah, fight or flight. When you're scared, when you're angry, those types of things. That fight or flight response, absolutely. Dopamine, remember, is the one we talked about, is involved in Parkinson's. As I mentioned, it's finding more and more. It's a very important central nervous system, a neurotransmitter involved in not just Parkinson's, but we're also finding in other types of social disorders and things along those lines. So dopamine is very, very important that way. Some larger neurotransmitters are some neuropeptides like endorphins. What kind of things give you endorphins? Is endorphins a good thing or a bad thing? Really? It's a good thing, absolutely. Endorphins are typically help you feel a little bit better, associated with things like exercise can cause endorphin release, love, chocolate, all of those things can do that. Right. Substance P, on the other hand, you may not have heard of. And substance P is the primary neurotransmitter in our pain pathway, hence the name substance P. And then remember, as I mentioned, our pain pathways are some of our least understood pathways that we understand, but those are an important neurotransmitter in that process. There are also some wonky things that can be neurotransmitters. ATP can be a neurotransmitter. Some gases like nitric oxide. Notice I said nitric oxide, not nitrous oxide. Nitrous oxide is what you get when you go to the dentist's office or that rave on Friday night, right? But again, if you go to the dentist or that rave on Friday night and partake in that nitrous oxide, one of the things you may notice about someone who does that is they typically get very flushed, very red in the face. Because what happens is that nitrous oxide affects you in a similar way that nitric oxide does. It's a vasodilator. We use nitric oxide to control the diameter of our blood vessels. So when you take it in that dentist's office, the blood vessels dilate, bringing the blood to the surface, and you typically get that very reddish coloration and that heat coming to the surface of your skin as a result of that. So I mentioned your book's got a nice table. There's over 100 ones that have been found, but these are some of the more common or more interesting ones that you will find in the nervous system. All right. Questions on that? All right, excellent. That is all I have for lecture today. Like I said, we have some lab catching up to do. So that is all we're going to do from a lecture standpoint. Let's go ahead and take our first break. We will come back at 9:15, and at 9:15, we will start working through our gross anatomy handout. We started hitting some of the key structures in that one. We'll continue our discussion and work our way through that. So we will restart at 9:15. And I will start the recording at that point. All right, any questions? All right, see you in 15 minutes. All righty, let's go ahead and get started. We are working our way through the gross anatomy of the nervous system handout that you should have. So hopefully you have that available so we can be talking about it. Notice we had started, again, a lot of this in the last class, so we'll quickly go through all of those things. Again, we're emphasizing differentiation of the brain regions into these cephalons. There are 5 main cephalons, all based on the development that happens and occurs early in development. It's not like you have to memorize this picture, but you do need to be able to recognize the cephalons in the mature brain. And here they've got it color-coded for us. As I mentioned, the big, largest component of the brain, the cerebrum, is the telencephalon. The telencephalon forms the cerebrum. Diencephalon stays a region called the diencephalon. Mesencephalon stays a region called the mesencephalon. The metencephalon is a region that becomes two big enlargements, the pons anteriorly and the cerebellum posteriorly. And the myencephalon becomes the region that is the medulla oblongata. And so again, if you, these three can be a little confusing, just remember they're in alphabetical order. Mes, met, my encephalons. So those are the cephalons. The cerebrum, as I mentioned, is the largest, most elaborate part of it, but it can get a little confusing that way. So here we see in a sagittal section, here is that telencephalon. Here is the diencephalon. Here is that narrow mesencephalon, which remember we also sometimes refer to as the midbrain, enlargement of the pons and the cerebellum, and the myencephalon, which forms the medulla oblongata. Lastly, we have this picture here where the cerebrum has been peeled off. And so we just see the diencephalon, mesencephalon, the pons, cerebellum's been removed to the met, and the myencephalon, which becomes the medulla oblongata. We also talked last time about the difference between gray matter and white matter. Again, remember, gray matter is going to be the cell bodies and the dendrites. They are going to be where we process the information, we make the decisions. And in our central nervous system, they form two structures. The outer superficial structure, which is the cortex, this whole big gray matter sheet that has all the folds in it, is crumpled up into a ball so that it fits in there, is the cortex, whereas these small clusters inside of the white matter are known as nuclei, nucleus singular, nuclei plural. White matter forms the pathways for information, and again, in our central nervous system, we refer to those as tracks. And notice in this illustration, you can see that there are different types of tracks. I'm not going to hold you responsible for the names of the different types of tracks, but I do want you to see some examples so you can understand how this works. So notice they're very close fiber tracks, what are known as the arcuate fibers, the arcuate tracks, because if you think about it, if you're feeling something with your fingers, what your index finger and what your middle finger are feeling, that information needs to be coordinated so you can make sense of it. If you're also trying to figure out what something is, you want to coordinate what you're feeling with your hand with what you're seeing with your eyes. So we have more longitudinal tracks that travel further distances for that. But also, if you're manipulating something with both hands, you need information from the right side and the left side to come together. And so for that, we need commissural tracks. And so these commissural tracks are bundles of axons that go from one hemisphere to the other hemisphere. Notice also we see really nicely that longitudinal fissure that we talked about. Remember, raised ridges of the cerebrum are known as the gyri, gyrus singular. The grooves are the sulci, sulcus singular. And a large invagination like this one is what is known as a fissure. And this one on the midline is the longitudinal fissure that divides the brain into two hemispheres. When we look at the hemisphere, we see that it looks a little bit like a mitten, where we have the thumb over here, and then we have the rest of the mitten over here where the fingers would go. So we have this anatomy here. Here we see it with some of the blood vessels and protective coverings over the top. Here we see that longitudinal view. And here we see that picture we saw last time where we left off with, showing those four hemispheres. Now, again, you all know what an amazing artist I am, so I spent a good hour and a half last night recreating this drawing as perfectly as I can, and there you see I did an amazing job of that. Okay, I did this in like 30 seconds during the break, but we still see that mitten part of it, and most importantly, it allows us to be able to distinguish the different lobes that we need to be able to identify. And again, we've learned the bones, so you know the lobes. Here at the front of the cerebrum is the frontal lobe. The thumb of our mitten is the temporal lobe. The very back is our occipital lobe. And actually, now that I've done this, I'm going to cheat. Move this up there. Move that down there. Occipital here in the back. And up here in the top is the parietal. Now, these are not just arbitrary in their definitions. There are actually some anatomical distinctions that allow us to recognize these 4.5 lobes. For starters, the temporal lobe is separated from the parietal and the frontal lobe by a deep invagination. And of course, being a deep invagination, it should be called a fissure. But as we know, anatomists hate us. So not surprisingly, they call it the lateral sulcus. So it is one of the anatomical features that helps us to distinguish the lobes. As we also talked about, there is a invagination, a consistent invagination found that basically starts at the, and I'll cheat and put it up here a little bit, at the top, because my picture is not the best, the top of the brain, and basically travels down towards the lateral sulcus. And remember, we said this was the central sulcus. The central sulcus, central sulcus is what separates the frontal lobe from the parietal lobe. As we also talked about, since there is a central sulcus, we know that there is a raised ridge here in front of the sulcus and a raised ridge here behind the central sulcus. And we gave names to those structures as well. The raised ridge in front of the central sulcus, what did we call that? Precentral gyrus. Excellent. And the raised ridge behind the central sulcus would then be the postcentral. Now, as we go back to the picture of the, so I guess I'll need to do this with a highlighter and make it as thin as possible. There is an anatomical structure that divides the occipital lobe from the parietal and temporal, but it is deep. So it isn't, and actually now that I've done that, I want to cheat and bring this out a little bit more. Actually, let's just get rid of the whole thing. I don't like you. All right, I'm going to go ahead and draw it as a line, even though it's really not. There we go. There is an anatomical feature that distinguishes the occipital from the parietal and temporal, but it is a deep structure that we can't see from the superficial surface, so it isn't there. So if we go back to the pretty picture from your textbook, here we see that anatomy we were just talking about. Here is that lateral sulcus. Here is that central sulcus, the precentral gyrus, the postcentral gyrus. All of this is the frontal lobe. All of this is the parietal lobe. All of this is the temporal lobe. And all of this back here is the occipital lobe. All right. With me so far? All right, excellent. So again, there isn't really any new information there. And if you think about it, that is everything at the beginning of your list. These structures, all of these things that we've identified so far are structures. And really, they're just anatomical features. They don't necessarily have a specific function per se. However, do portions of them have specific functions? Absolutely. So again, all these things that I've pointed out are just structures. So on your handout for these things, you could just describe them however you want to describe them so that you can easily identify them. Again, the advantage of using illustrations like this on the lab exam is that with it being color-coded, it makes it a little easier for you to be able to differentiate these things, right? If we look at it as something like this, especially when they have the labels, Is it possible to find the central sulcus? Absolutely, right? Again, it starts at the top, projects down towards the lateral sulcus, but without the labels, without the feet leaders, it may take you a few seconds to do that, and that's not my goal. My goal is to get you past identifying what the thing is so that you can figure out what it is that you're actually responsible for. So I will try to use images that make some semblance of sense. All right, now, everybody's brain is unique. However, one of the interesting things about brain anatomy is there are some similarities, right? Back in ancient times, I actually get to share this with you. Back in ancient times, and by ancient times, I mean the late 1800s, there was a neuroanatomist by the name of Corbinian Brodmann. Corbinian Brodmann was an incredibly patient individual. Obviously, in the 1800s, late 1800s, we did not have the type of technology that we have today, where we can throw someone in a functional magnetic resonance imaging machine and see how their brain lights up when they do different types of activities. They couldn't really tell what the brain was doing, but what he was able to do was look at individual brains. And so what he did is he actually started in a cat. He pulled out one cat brain and he examined it very thoroughly. And then he pulled out a second. And as he was looking at the two of these, one of the things he noticed is, you know what's interesting? Both of these brains have a raised ridge in this area right here. And then he picked out a third and a fourth and a 215th. And after looking at all 215 of these cat brains, he's all, yep, all of these brains have this one structure that is fairly consistent. Did he know if that structure did anything or what that structure did or anything along those lines? No, but he found that this was a consistent structure. While there are some differences in brain anatomy across these 315 cat brains, this is a characteristic that they all have in common. And that might mean it's something important. So I should give it a name. And since it was the first region that he found that they had in common, guess what he called it? Well, he called it Area 1. And then he found a second area that all the brains had in common. And guess what he called that? These are the easy questions, people. Absolutely. Area 2, and Area 3, and Area 4, and so on and so forth. As he went through these things, and I'm going to go ahead and cheat and remove that from there and come back to this instead, we have these what are known as Brodmann numbers. Because good old Corbinian Brodmann is the one who identified and described these first. Here we see some examples, Area 1 and 2 and 3 and Area 44 and Area 45. He found all these in cat brains, and then he looked at monkey brains and found similar structures, and then he looked at human brains and found similar structures again. Again, all these are identifying numbers for structures. But the fact that we're talking about them should tell you something about it. It turns out that the areas of the brain that are similar in structure typically are similar in function as well. Now, does that mean that you have to memorize that Area 4 for Brodmann is the premotor cortex or the primary motor cortex? No, absolutely not. I think of these Brodmann numbers as the numbers on an arm or leg model. If we were in the classroom, when we were going through the forearm, for instance, where the forearm can be more challenging, as you're looking at those, and if I was able to say that the flexor carpi radialis was number 17, was muscle number 17, then if you were all holding a model and looked at that model, could you easily find muscle 17 and know exactly what it is that I was talking about? Yeah, absolutely. So when I tell you that area 17 is the primary visual cortex, for the exam, do you need to know that the primary visual cortex is Brodmann's number 17? No. But if I have an arrow pointing to the very back of the brain there, and I ask you the name of that area, Do you need to know that it's the primary visual cortex? Yes. Do you need to know that that primary visual cortex allows you to perceive light? Yes. And so if I tell you that that is area 17 as we look at this illustration, does everybody know exactly what part of the brain I am talking about? This is the part where you all emphatically say yes. Still waiting. There you go, exactly, excellent. Yes, absolutely. So again, I will use these numbers to help us to identify clearly what parts of the brains I'm talking about, and I'll draw them on my pretty picture as well. Was it perfect? No. Every structure that he found didn't necessarily have its own unique function, but enough of them did that it was impressive enough that these are numbers that 200 years later we are still using. All right, questions on that? All right, so what we're going to do, as you can see for your illustration, is we are going, from your handout in front of you, for each lobe, we are going to identify 2 or 3 areas or regions. That is the term I will use on the exam, but let's be more specific of what I'm talking about. These are areas or regions of the cortex. Right? This is the gray matter on the surface, that superficial gray matter that we are identifying. And for these 2 or 3 areas or regions we identify, not only do you need to know the name of these areas, but you need to know the function. All right. So again, let's take one more picture before we do that, because there's one other thing where I wanted to mention. Notice now as we cheat and go back to this coronal section, notice we can see some of the anatomy. We focused here on the gray matter and the white matter, but notice also as we're sitting here, we can see the longitudinal fissure that goes down the midline, separating it into the left hemisphere and the right hemisphere. Notice also we can see the lateral sulcus. Remember, the lateral sulcus separates the parietal lobe up here from the temporal lobe down here. So we can also see that lateral sulcus. And notice how deep that lateral sulcus is. So like I said, it probably should be called the lateral fissure, but as we know, anatomists hate us. So this is the lateral sulcus, and this is the longitudinal fissure. All right, questions on that? All right, so let's start here. Clear my drawings there and switch back to my pretty picture. And we will talk first, and let's use my highlighter for this, about the frontal lobe. There are 3 areas in the frontal lobe you are going to be responsible for. The first one is basically this entire anterior region of the frontal lobe, what is known as the prefrontal prefrontal cortex. The prefrontal cortex is the part of the, oh, hold on. I need to find something. I don't like it. This is better than nothing. That's not the best. Sorry, I apologize for not getting this beforehand. I am so used to doing this in the classroom where we have our half head, our sagittal brain, that we have the chart available, and I don't know where to find an easy example of it. Let me check one more place. Ah, that works. That's what I want. All right, perfect. All right, excellent. Prefrontal cortex. Prefrontal cortex is the part of the brain that is responsible for logic, reason, personality, emotion, things along those lines. How do we know this? How do we understand that? Like I said, nowadays, we can throw somebody in a functional magnetic resonance imaging and non-invasively see blood flow in their brain and see what their brain does. So how do we know that the prefrontal cortex allows for that emotion and that cognitive thought and decision, personality, and all those types of things? How do we know that? I'll wait.
Is it through like maybe people had accidents and we also have the machine that lights up the brain when people have
Right.
emotions and stuff like that?
We have the machine now, but we didn't before. So you're right. Absolutely. It was primarily by injuries, right? One of the best things that happened to the field of neuroscience is the development of helmets. Because before helmets, young men went off to war, and what happened? What happens when young men go off to war? Especially if they don't have a helmet? These are the easy questions. They die. No, they die, right? They die. Absolutely. However, once we got them helmets, then they only came back injured, brain damaged, right? So the helmet helped to keep them alive. So they were only missing part of their brain instead of all of their brain. You lose all your brain, you're dead. You only lose part of your brain, you don't necessarily die. So they'd roll some guy in, the back of his head would be blown off, and he wouldn't be able to see. And we go, oh, hey, that back of the brain must be what's responsible for our vision. Well, our prefrontal cortex is the same way. There are many classic examples, but probably the most classic example that many of you heard of is an individual by the name of Phineas Gage. Anyone here know who Phineas Gage is? Come on, some of you must have taken a psychology class where you heard about him. Vaguely. All right, well, you guys have all lived way too sheltered a life. Phineas Gage was a foreman on the railroad. Yeah, there you go, exactly, the pole dude. Exactly, pole through the head. He was a foreman on the railroad. And the foreman on the railroad is a very responsible individual. And one of his responsibilities is dealing with the dynamite. Because if you're building a railroad, you're trying to get a train from one location to another, and you're trying to do it as quickly as possible. So if you come up across a small hill, do you waste time and energy and resources to divert your train tracks around that hill? No, you blow up the hill, flatten it out, and you keep your track going straight. The way they do that is they drill a large hole into the rock, and then using a tamping rod, a 10-foot metal pole, the foreman is responsible for tamping, hitting it with a hammer to drive that dynamite down into the hole so that when it blows up, it maximizes the damage that is being done. Well, one day, good old Phineas was doing his job of tamping that pole down, and the dynamite went off. And when the dynamite went off, the pole hit him in the forehead, came out the top of his head, through his head, 40 feet into the air, knocking him back 20 yards. And shockingly, he did not die. But his prefrontal cortex was destroyed, and his personality changed tremendously. As I mentioned, as a foreman, he was very mild-mannered, very even-tempered. If you go and you read some of the reports, he went from this mild-mannered, even-tempered individual to the type of man, and I quote here, that would drink in the afternoon and curse at women. It completely changed his personality. And that and other research, animal research, has shown similar types of things where this prefrontal cortex is associated with personality and associated with aggression. Well, at the time, this was a tremendous finding, right? Because now we had a location for where your personality was. So if we didn't like your personality, maybe you had an abnormal personality, right? Maybe you were a male who, wrongly liked other males. Maybe you were a female who was of the opinion you were equal to a male, right? These are all horrible ideas for an individual to have. And if something like that occurs, you got locked away in a sanitarium. Was that sanitarium so that you could get better? No, basically you got locked up into a sanitarium so that you couldn't harm yourself or anybody else. And you basically stayed there till you died. But now we had this massive new piece of information. Your behavior, your personality is located in the front of your brain. So what if we could modify that some way? What if, and here is that chart from the classroom, we were to take a narrow, thin metal tube. And that narrow, thin metal tube had a little bit of a slit in it. And what would happen is they would insert that narrow metal tube. Often they would do it, this isn't going to work. Often they would do it, here, I'll do it this way. In through the nasal cavity, where they could break it up through the cribriform plate. As I mentioned, it had a slot in that narrow metal tube. And so what they were able to do is they would shove a piece of razor wire into that tube, whoops. And so that when they pushed it, it would bulge out that slit. And then what they would do is they would rotate that razor wire around in the prefrontal lobe, destroying that nervous tissue until your behavior changed. And yes, you were awake when this occurred. Not surprisingly, the people were apprehensive and nervous coming into these procedures, and they would continue to do this till you weren't nervous anymore. Exactly, as Madison pointed out, this is what we call a prefrontal lobotomy. Again, hopefully you appreciate the barbarianism of what we are talking about here. Right, I want to make sure my sarcasm is coming across with these comments on this. This was a horrifying behavior that these people were doing it, and it was far worse than that. There were many doctors that would roam the country going to sanitariums doing these things. One of the most famous ones was a doctor by the name of Watson. Watson actually would have two of these cannuli, that's what they called these tubes, in holsters on his side. He would go to local sanitariums as he would travel the country, and he would compete against local doctors for speed, right? He'd be able to use both in both hands through both nostrils at the same time to be able to do this, and he'd compete for speed with other individuals. Again, horrible, horrible, horrible activity. So why in the world were they doing this? Well, because as I mentioned, as wrong as their behavior hopefully is perceived to be now, the idea that we may be able to modify people's behavior was a very powerful idea. Like I said, up to that point in time, if people had what we thought to be abnormal thoughts or abnormal actions, they basically got locked up into a sanitarium and they were stuck like that for life. The allure of the potential of being able to fix somebody so they could go out and live a normal life again is really powerful. And did this improve some people's behavior as a result of that? Yeah, there were some people who were improved by this, right? But other people weren't improved. Other people got worse. And we're not even talking about the infections and the drainage of cerebrospinal fluid into the nasal cavity and all the other medical issues that went along with that. But there was a small section that were improved by this, and so it helped to drive this process for many, many, many years. Thankfully, this isn't done anymore because people finally realized the barbarianism of what they were doing and they stopped it because they realized it was wrong. Is that why they stopped doing this? I'm going to stand up here in my soapbox and rant. I expect you guys to respond. Did they stop this because they realized it was wrong and it was amoral and we shouldn't do it anymore? Is it illegal now because people realized it was wrong and immoral? No, they stopped doing it. And I will warn you, it's not actually illegal in all states. There are some states where technically it is still legal. No, the reason it stopped is because, not because it didn't work, not because it was wrong, not because it was amoral, not because it was damaging, but because they found something cheaper and easier. Why should I bother having to pay a whole ton of money for this specialist who's going to fly across the country to come here and change the behavior of my patients if I can just give them this cheap, easy pill instead? I don't know what the pill does. All I know is that it changes this person's behavior. Does it change everybody's behavior the same way? No. I have 3 patients, both having the same abnormal behavior, so I give all 3 of them the same pill. One person gets better, one person it has no effect, one person gets worse. Great. So I'll give a different pill to the person who isn't doing better. I'll give 2 pills to the person who got worse. I'll just keep mixing and matching all these different medicines that I don't know what the hell they're doing, but I do know they're changing their behavior until they behave in the way that I want them to behave. What we are doing with medication is just as barbaric as what they were doing with that prefrontal lobotomy. We don't know what most of these medicines are doing to individuals. Have it gotten better over the past 20 years? Yes. But are we still prescribing way too many medications that we don't know what they're doing other than changing people's behavior to what we think is appropriate for the situation? Absolutely. So I truly hope that 50 years from now, we'll be laughing at the medication of patients the same way that you and I are laughing at these prefrontal lobotomies. It all comes from a good spot. We want to improve people's behavior. We want to make people better, but we have to be careful about how we do it. All right, off my soapbox. Questions on that? All right, let's talk about the second region in our frontal cortex. Change the color slightly so that we can see this. There are 3 distinct gyri that kind of line up here in the frontal cortex. Again, I've drawn a simplistic view of this. Let's look at the pretty picture from your textbook. Notice here, and I'll use black, these 3 right here. Notice this happened to be Brodmann's areas 44 and 45. This is an area that is known as Broca's area. Why, of course, is it called Broca's area? Because good old Bob Broca was the first person who described it and discovered it. Absolutely. It is also, though, and this I think will maybe hopefully help us to understand a little better, it is also known as the speech area. And what do you think the speech area does? Not your question. It allows you to produce meaningful speech. Absolutely. Right? This is the area where we produce meaningful speech. Our ability to produce speech and meaningful speech and understand speech is a highly sophisticated process. Right? Speech is a specialization that has allowed us to communicate, and our brain has been very, very specialized to doing this. And this is one of those areas. Now, one of the interesting things about this is, remember, we talked about the concept of lateralization. Lateralization is this concept where what is happening on one side is different from what is happening on the other side. Broca's area for most individuals is actually located over here on the left hemisphere. On the right hemisphere, for most people, the area that would be Broca's is typically more associated with math computations and things along those lines. But most people, especially most, over 90% of all right-handed individuals have their speech area over here on the left side. Now, again, one of the ways we fully understand how a region works is when that area is damaged. And damage to Broca's area, or that speech area, causes a condition we call aphasia. Oops. There you go. Aphasia. Broca's aphasia is a condition where you have trouble producing meaningful speech. Instead of saying, Billy and I are going to the store to get milk, you would say, Billy, store, milk. It doesn't have the prepositions, it doesn't have the cadence, it doesn't have the speech. Now, obviously, a complete ablation means that you would have difficulty producing any speech, but there can also be minor problems with this area as well. And minor problems with the area may produce some types of stutters as a result of that. Out of curiosity, does anybody who's either had a stutter or knows someone who has a stutter know one of the tricks people with stutters use to get around those stutters? What do they use, Madison?
They typically use either like keep it simple words or they pause a lot before they talk.
Okay, absolutely. Changing the cadence is definitely one of the ways. Another way that they can get around it is actually by speaking in a sing-songy voice.
Yeah, it's like a rhythm.
Yeah.
Yeah.
I are going to the store to get some milk. And again, we laugh, but one of the interesting things about our speech area is it really is specific for speech. Singing is a completely different process. Now, I know you probably haven't thought of it these terms, but I know you're fully aware of it, right? Because how many people here have heard Adele sing? Most of you. How many have heard Adele talk? Yeah. When she talks, you can barely understand what she's saying. It's like she's talking through a mouthful of marbles, right? She has a very huge, thick accent. But does that accent come out at all when she is singing? No, because singing is a completely different process. So you see that with a lot of singers. The way they talk and the way they sing are dramatically different because they're actually using different parts of the brain. And so that's what they teach these people who have stutters. If they can talk with more of a sing-songy to their voice, they're able to avoid that Broca's area and it can actually help them. Tourette's syndrome. Tourette's can be associated with this area, but it is also other areas that can have, typically Tourette's is associated with more than just speech. That speech impediment is one of the byproducts of that neurological disorder. All right. Questions on that? All right, the third area, so let me grab my highlighter again and switch to our third red. The third area that we are responsible for happens to be located on one of the structures we are responsible for. We are responsible for knowing the structure that is the precentral gyrus. And the structure that is the precentral gyrus is the area, or contains the area. So again, let's emphasize this. The precentral gyrus is the structure. But on it, there is an area, and that area is known as the primary motor cortex. And guess what the primary motor cortex does? Movement, and more specifically, you're absolutely motor skills. It controls, if we want to be precise with our definition, it controls contralateral skeletal muscle. All right, I like that. Let's write that down. Controls contralateral skeletal muscle. So my primary motor cortex over here is how I move my right hand or shake my right arm. All right, it's contralateral skeletal muscle, voluntary control. What's cool about this area is there is actually a map of the body on this precentral gyrus. So for instance, way down here is movement of the head, and then up here is movement of the hand, and then up here is movement of the arm, and then up here is the trunk, and then up here are the legs. Now, of those 5 parts of the body, which one's the biggest? What's your biggest part of your body? Your arm, your leg, your trunk, or your head? What do you think the largest part of your body is? And this shouldn't be a hard question. Legs are definitely pretty large, maybe trunk. Those probably are 2 of the bigger areas of the body. I like legs, I like trunk. Those are good answers. However, in this area, the largest representation is actually of the head and the hand. Your biggest representation is this: the head, especially the lips, the hand, especially the thumb, have a huge representation in here, because those are the areas where we're most dexterous. And then less of the arm, very little of our trunk. There's only so much movement of our trunk we do. So the representation of the cortex is more representative of how we use the parts of the body than the actual size of the parts of the body. So we have this fancy map on here. And again, you may not have thought of it in those terms, but you're aware of that because there is a blood vessel that, whoops, that's a horrible, let me undo that. There is a blood vessel that travels down your central sulcus. And if that blood vessel happens to get blocked in a particular area, blood flow to that part of the brain stops, and that nervous tissue can be damaged. What do we call that condition? When blood stops going to a stroke, absolutely. And a result of that stroke could be I can no longer move my hand, or I can no longer move my arm, or I can no longer move one side of my face from that stroke that occurs. Interestingly, on the parietal lobe, right across on that postcentral gyrus, is the first area of our parietal lobe. Again, the postcentral gyrus is the structure. Here we have the area on that structure, and that area is the primary somatosensory cortex. What do you think the primary somatosensory cortex does? Yeah, exactly. This receives contralateral touch of the skin. So when I touch something with my right hand, it goes up here to the primary somatosensory cortex of my contralateral brain. And we have that exact same big, huge map where there's a huge representation of the head and face, a huge representation of the hand, especially the thumb, and then a smaller for the arm, and a smaller for the trunk, and the legs, and so on. We actually have that exact same map mirrored itself essentially on that cortex. Because again, if you think about it, when that stroke occurs, typically the entire region is damaged. Now, not always symmetrically, right? So you may lose touch and feeling touch and movement of the hands, but only movement of the arm. You may still be able to feel on the arm. So it's not going to be perfectly symmetrical, the damage, depending on how severe the damage is. But typically both movement and touch are affected by those strokes because that blood vessel goes right down here, down the center. All right. Questions on that? All right, there is a second area of the parietal lobe we are responsible for. So let's switch to that. And it's in the parietal, although it can sneak a little bit into the temporal. It's kind of in this area here. And this area is what is known as Wernicke's area. Of course, why is it called Wernicke's area? Good old Bob Wernicke was the first one who identified and described it, absolutely. Or what is also known as general interpretive. Now, let me show you a couple quick pictures of this before we talk about what it does. Coming back here to our book, notice here, this dotted region that includes some of 22, but other areas as well. Notice this image here, it's 22, but also 39 and 40. Although here's the best picture. Here, notice here, they have given us, there's Broca's area, the speech center, and here's Wernicke's area, that general interpretive. So really, the easiest way to find Wernicke's or the general interpretive area is to follow the lateral sulcus back. And when you follow the lateral sulcus back, that is where you'll find Wernicke's area, which is the general interpretive area. Now, Broca's area is where we produce speech. Wernicke's area is where we comprehend speech, right? This is where we make sense of what we hear and what we say. Notice, if I have damage to my Broca's area, and you tell me to raise my left hand, I know that's my right hand, but you get the idea. I can still hear what you're saying, understand what you're saying, and follow directions. That isn't always the case with Wernicke's area. Again, if Wernicke's area is damaged, and this is another lateralized area, in right-handed people, over 95% have it on the left side. For left-handed people, 75% have it on the left. About 15% have it on the right. And about 10% have it bilateral on both. Now, we'll talk about what's on the other side in just a minute, but let's talk about this Wernicke's area. People who have damage to this have a condition that we call Wernicke's aphasia. Let's take a look at an example of this.
You know, boom.
What's this?
You know, the big anchor right there. End of the football.
Can you guys hear that? It's a little muffled, so turn up your volume. I'll be quiet here and let's see if you guys can hear it. Just do your best that you can to hear it. If not, you can always go back and look it up on YouTube. That's where I found this, just by searching Wernicke's aphasia. But let's try to listen to it.
They're afraid, you know, boom.
What's this?
You know, the big anchor right there. End of the football.
End of the football?
It influenced the football right here. The cause that it comes in, see?
Yeah.
The end of it, see? Yeah. Oh, we have all of these, see?
I don't understand.
Well, it's Well, it's pliable in the gut. See, you don't understand it, but it flies in the long tree comes from the head, and it comes soars from the country out here. See, all the under it. See, it comes up and finally come out here and goes out. See, get it?
Sort of.
Yeah, it comes out here. See, it comes out here, here, you and here,
Uh-huh.
and it comes in, and it comes out, and bing. That's a good fly throw. See.
That's a good what?
That's a good flow goes the fly, or see it throws cold or it gives or kicks out or bing or whatever it is. We has no football about it out. It's just pulling, see, but they feel live. Oh, I guess you meant you have bed, see, I don't know.
Where do you live?
Where do you live? You live? You're up here, see.
Where do you live, Doc?
Where do you live?
Where do you live?
Where do you live is, well, if there's a run through here, around here. You?
You used to be a dentist.
Yes,
Where?
right here. the rest of the version, yes.
Yeah. How many years were you a dentist?
How many? Holy Christmas. Oh, about 100 and, let's see, 130, about 40. No, that's what that is. One plus 5. Wait a minute, no. It's a pretty close to get to this one day. It's about every year off, see. It's claws. It goes for about over 30, see, or 130 for down, see, in front of out for it goes straight against her, and then it comes back to the against her 4, and then goes out for the rest of the time. The third is not the servant, but the soldiers, where they have the soldiers of the same birds and the suburbs and all the first suburbs and watch out in the short side, and they cross with the sugar 2 hours, 2 high or low. Wow, God, these are terrible. Wow, these are terrible words.
These are terrible words.
They sure are.
Hard?
Yes.
What you're saying, you mean?
Yeah, that's what they do. They actually go them a lot harder. It's when they are in hard go. And then when they go out, finally they quit, see?
So notice, if these two were sitting at a Starbucks behind you and you weren't really paying any attention, it just sounds like they're having a normal conversation. It isn't until you actually start listening to what he's saying that you understand that what he's saying isn't necessarily making a lot of sense. He has all of the normal cadences to speech. His speech area is not damaged. He's able to produce normal speech, but the speech doesn't necessarily have meaning. Some of it does. He doesn't have a complete aplasia, but obviously he is having difficulty explaining what it is. Now, I've watched this video many times, so there's a lot of parts of it that I've subtly picked up on and what he understands and what he doesn't. She asks him, you know, you were a dentist, yes, where? And he points in his mouth and laughs, so obviously, you know, pointing to his teeth. But notice also, she asks him, where do you live? Now let's switch that around. Here, you and I are sitting here in the classroom, and I walk up to you during the break, and I ask you, where do you live? Right? Now you're going to have to think about that for a minute. And as you think about that, you're like, where do I live? Right? You're thinking about the question, do I really want him to know where I live? Right? Do I want to give him that information? Do I lie to him? Do I just tell him no? Do I tell him I don't have a home? Right? You're thinking about that, and you turn it around. I say the words, where do you live? And you turn them around while you're thinking of them, where do I live? Right? Because you've taken that question, you understand that question, and you're turning it around. She asks him, where do you live? and he repeats it back to her verbatim. Where do you live? He's not interpreting the words. He's not able to turn them around. And he's not able to answer the question for her because he doesn't understand the question. He says what she says word for word, right? And then she asks him how long he's been a dentist. And clearly he spends a lot of time trying to answer that question. Now, it appears to me, as I've been watching this many times, that one of the reasons he's having difficulty explaining this, because what apparently happened is he was probably a dentist for a period of time and then had to go away to war, and then came back from war and was a dentist for the rest of the time. Because he's talking about the soldiers and going around the short side and the uniforms and all those types of things. So I think that's one of the reasons why he's having difficulty counting, is because the time was broken up that way. But it takes him a very long time, and he never really does come up with a correct answer to that. However, the thing that I think that's most interesting about that video is what happened at the end there, is that you see some of his frustration. These are horrible words, right? And she's all the horrible words, and and he kind of explains, and again, he's not able to verbalize it, but what he's saying is, I'm able to think of these things, but then when the words come out, they quit. He can process the information, he knows what he wants to say, but he's not capable of saying it. So obviously, he, again, shows some frustration at that point with his situation. How much these people understand their situation is hard to determine, because obviously if they can't communicate to them, but clearly he shows some understanding that he is having challenges getting these words out.
Is this what happens to people with Alzheimer's?
No, it is not. Let's say it this way. It is not a symptom of Alzheimer's. Alzheimer's is a degenerative neurological disorder. Now, is it possible that that degenerative neurological disorder could potentially happen at or near Wernicke's area? So someone with dementia or Alzheimer's could show some symptoms associated with this? Yes, but it's not, but it isn't directly because Alzheimer's. There's plenty of people who have Alzheimer's who don't necessarily have this component of it. It would really depend on what portions of the brain are being deteriorated by the Alzheimer's or the dementia.
Okay, because this is kind of like how my grandpa communicates with this.
Right.
So
Yes. So some of that can be challenging. Yes. And again, it depends on where the degeneration has happened. So is it possible that Alzheimer's can affect this area? Yes, but it isn't a requirement of Alzheimer's, if that makes sense. All right. So again, hopefully you guys were able to perceive some of that. I don't know how well that came across the video, but if not, like I said, I encourage you to go watch it on YouTube. That's where I found it. And I think, again, one of the nice things about, well, okay, it's not nice, but one of the powerful things about this is often we can really understand what an area does by seeing the deficiencies. And that brings us back to the last point. As I mentioned, especially if you're right-handed, over 95% of you have Wernicke's area over here on the left. So what the heck is over here on the right? And it turns out, and let's cheat and go back to my pretty picture. On the right, basically, people have spatial awareness. Awareness of the contralateral side. It's a spatial awareness. Again, this is one of those cases where people have an injury to this, a deficiency to this, and it can cause some really unique and interesting characteristics. Someone who has damage to what would be Wernicke's area, the spatial awareness on the right side, you ask them, you show them a potted plant, and you ask them to draw that potted plant, and what they will draw is this. They completely ignore the contralateral side because that damage to the spatial awareness on the right side makes them unaware of everything on the left. So when you ask them to draw that potted flower, that potted plant, they only draw the right side of it. I had a student 2 years ago whose boyfriend was in a car accident and had a traumatic brain injury to the right side of his spatial awareness. And he, when he got dressed in the morning, would forget to dress the left side of his body. When he was in the shower, he would not wash the left side of his body. He'd be sitting at a restaurant and get angry at the person next to him for getting too close to him and getting in his space. Get away from me, buddy. What are you doing here? Right? He wouldn't recognize the left side of his body, would forget to dress the right side of his body, wouldn't wash the rest side of his body. They ignore things basically from the left side of the world. All right. Questions on that? All right. Let's talk about vision. Again, I am completely 100% biased. I am a visual neuroanatomist by trade. I got my PhD studying the visual system, and I love the visual system more than I love my children. All right. But my bias aside, we are visual organisms. Over 60% of the information we take in from the world around us is visual information. Over 40% of our cerebral cortex, of our brain, is involved in processing visual information. We are visual organisms, and vision is vitally important to us. All right. And it all starts in that area we talked about way back here at the very, very back of the brain, that area 17. And again, you don't need to know that it's area 17. I'm just writing that there to remind you when we look at the picture. This is the area we call the primary visual cortex. And the primary visual cortex is the part, as the name would indicate, where we process, or where we receive, let's say it this way, we receive visual information. This is where we perceive light. So if your area 17, your primary visual cortex, was damaged, what would you see? Nothing, exactly. You would not be able to perceive light. You would see nothing. All right, it'd be like basically having your eyes covered all the time. It would just be completely black. You would see nothing. Now, hopefully none of you have had damage to your area 17, but maybe you've fallen down a flight of stairs, or maybe you have fallen in some other instance, or maybe you've been punched in the face. And as your head rocked back, one of the things that happens is, even though your brain has that cerebral spinal fluid surrounding it, maybe the back of your brain got pushed up against the back of your skull. And what happens when that occurs? Well, okay, a concussion absolutely can be one of the things that can affect, that can be more traumatic than that. What else can occur? True, you can black out if you lose consciousness as a result of it, but those are a little more extreme. I'm thinking a little less extreme. Blood brain, again, all these things are horrible things. I'm thinking not necessarily that severe. Yes, all of these things can occur and they're very severe, and that's a very bad thing. But if you get hit in the head, right, and your brain bounces back, right, on the cartoons especially, do you really see black? Or do you see light? You see stars, right? On the cartoons, you always see the stars circling around their heads. You see stars, absolutely. You see bright light. When your head gets pushed, well, your occipital bone gets pushed into your occipital lobe, into that visual cortex, we often see a white light. So you see sparks, or you see a white light, you see those stars as a result of it. That's where that comes from, is from that pressure on the back of the brain, and you see those stars as a result of it. So again, let's cheat real quickly and go back to the pretty picture. Beep, beep, beep. There's a good one. Here's a good one. So again, very, very tip, very back of the occipital lobe is our primary visual cortex, which again is where we receive the visual information, get the basic information, light versus dark. But then we need to process that information. Oops. We need to make sense of it. And the way we do that, the way we process it and make sense of it, is with what we call the association cortex. As it turns out in our nervous tissue, and I'm going to exaggerate this not so much, but they're basically, oh, hold on, I don't want to use the same color. That one the one I didn't use? No, that's the same. Are these both looking the same? Oh, there you go. Okay, hold Excellent. All right. There are these big, huge pathways that actually go out into the parietal area and also come down into the temporal area. These big, huge areas of visual cortex where we process it. And there are basically two main pathways. Those pathways are the where pathway and the what pathway. Now, some of the information is pretty basic. Let's go ahead and I'm going to cheat and use yellow because I've run out of greens. Nope, that's not yellow. That's yellow. This area that is right outside area 17, and again, I'm giving you the number just because it helps me to identify what we're talking about here, area 18. So let's cheat and go back to the pretty picture and look at this. Notice this area right here, area 18 is part of the visual association cortex, where we make sense of that information. And area 18 is actually where we perceive color. So notice in this case, if you had damage to area 18, you would be able to see light. You would be able to see black and white and different intensities of light, but you wouldn't be able to differentiate colors. So the whole world would be like a huge black and white film noir. Now, I'm not talking about things like red and green color blindness. That is happening in the retina, and if we get to a chance to talk about the sensory system, we'll talk about that there. But this is from a cortical standpoint where we make sense of color. Again, one of the ways we can understand what these areas do, and they get more and more sophisticated as they go up, is by understanding what happens when there's damage to them. The where pathway allows us to both see where things are in space and also see how they move. There is an area in this where pathway called MST. This is a region of the brain. I'm not going to hold you responsible for this name, but I just want to give you an appreciation and an understanding of what it does. So great question. So for the color blindness, in that case, what it is doing is it is changing. Typically, color blindness is due to lacking certain photoreceptors in the eye. And so if we can bend the light in a way where it's easier for our eye to differentiate the colors, it can make it easier to see. So in some types of color blindness, that can be used to help to differentiate the colors that way. So yes, but that's not what we're talking about here. Here we're talking about cortical issues where it would affect both eyes equally, and so it's not quite the same thing. But it is our perception and understanding of color. MST, and again, MST is not a term you're going to need to know, but I'm just using this as an example, is an area of your brain involved in optic flow. If you think about it, as you move through space, oh, hey, I can give you an example. As you are moving through space, and as you're moving forward in space, the world is coming towards you. As you rotate, the world changes as you move towards it and things along those lines. So we're able to see that change or that flow of the world, and it gives us a sense of where we are in space and how we're moving through that space. Well, people who have damage to MST don't see that optic flow, right? How many of you have ever been in a room with a strobe light before? Anyone ever been in a room with a strobe light? Excellent, at least one person has. Several of you, excellent. If you and I were standing in a room with a strobe light and I was waving back and forth, would you see the continuous movement of my hand? No, you'd see my hand here, and then suddenly it would be here, and then suddenly it'd be here, and then suddenly it'd be here, and suddenly it'd be here. You wouldn't see the movement. You basically would see a bunch of static images. Well, that's what it's like. You are constantly at the rave if you have damage to MST. You cannot see movement. Can you drive a car if you can't see movement like that, if you can't see optic flow? No, absolutely not. Heck, can you pour yourself a cup of coffee? Coffee's here, coffee's there, suddenly it's overflowing. No, you can't. Now, Does that mean people with MST don't drink coffee? No, of course they do, because people are clever. Any idea how a person with MST could actually pour themselves a cup of coffee? You put your thumb in the mug. Now, if you think about it, if you put your thumb in your mug, you could pour yourself a cup of coffee with your eyes closed. Just pour it till you feel it with your thumb, and then you stop. So you could technically do it, but that's what it's like. People with damage to MST don't see movement, don't see optic flow. They are constantly living in a rave. Conversely, the what pathway helps you to identify what things are. There are cells there that are very unique, very discrete for just identifying certain things. There's actually an area here called IT. And again, you don't need to know the name IT. But IT is an area that allows us to differentiate and identify objects. Probably the classic example of this is there is an individual who has a damage to his IT region. I got to get rid of my drawing there to do this, where he can see things just fine. And one of the classic experiments with him, what they did is they showed him a glove. So let me draw my glove here real quickly. They showed him a glove. 3, 4, 5. And he was able to describe it perfectly, that it had these extensions on it. It happened to be colored with alternating yellow and blue stripes. And he was able to describe all of that. He could describe the colors, he could describe the stripes, he could describe the characteristics of the glove. But then when they asked him what it was, he couldn't answer the question. He sat there and looked at it for a while, and because it had these extensions coming off of it, he thought, oh, you know what? It must be a coin purse, right? One of those is for pennies, and one of those is for nickels, and one of those is for dimes, and one of those is for quarters. They then hand it to him, and when he holds it in his hand, he's finally able to make the connection and figure out that it is a glove. And it doesn't just work for objects either. He is married. However, if his wife stands perfectly still, he does not recognize her. The second she speaks, he can recognize her and knows who she is. Sometimes when she walks, the movement of her allows him to recognize her. But if she is standing right in front of him and not moving, he does not recognize her. Fascinating individual. There have actually been several books written about him. One of the most famous ones is The Man Who Mistook His Wife for a Hat. So again, it gives you an appreciation and understanding of how complicated this association cortex can be. There is right, we went from primary cortex where it's just light. Is there light or is there not light? Yes or no. To incredibly complex types of processing, like optic flow and item identification. So this visual association cortex is basically what allows us to make sense of the light that we see. And it doesn't just work for vision, it works for hearing as well. However, in hearing, it occurs in the temporal lobe. Basically right here, highlighter, brown, where the central sulcus points, basically right where the central sulcus points, right here on the temporal lobe, is our primary auditory cortex. Primary auditory cortex is where we receive the basic auditory information, sound information. But again, we then need to make sense of that sound. So, whoops. So hold on. No, I want that to be brown. Did I screw that up already? I did screw that up. Oh. So I'll change colors. Purple. Around it here, this area here is then, of course, going to be our association auditory cortex. And that is where we make sense of the auditory information. Primary auditory cortex is where we hear a sound. The association cortex is where we realize that it is a sound that is alternating between two frequencies, and it is increasing in amplitude. And that tells us that, oh, it is a siren coming towards us. I need to get out of the street. So we receive the basic information, and then we process that information and make sense of it. All right, and if we go back to our pretty pictures, because I've made a mess of this here, notice areas 41 and 42, this basically right below the central sulcus. And notice again, a little bit of artistic, again, central sulcus comes down, and then right here is that auditory cortex, primary and association auditory cortex. Primary and association visual cortex. All right, so there you go. Just that simply, we have identified the 4 lobes of our cerebrum, the structures of our cerebrum, and the areas of these 4 lobes that you are going to be responsible for on the exam, on the lab exam. So on the lab exam, if you think about it, I can point to, I can have a nice big arrow pointing right here. That's not an arrow. I have a nice big arrow. That's not big. I can have a nice big arrow pointing right there. And basically ask 2 questions. Identify this area of cortex. Identify the function of this area. Notice also, I could have a nice big arrow pointing here. Actually, I lied. There are 4 questions I can ask back here. 4 questions I can ask with this arrow. Identify the area, primary visual cortex. Identify its function, perceive light. Identify the lobe, occipital. Identify the cephalon, telencephalon. Notice for my second arrow, I can ask those exact same questions: cephalon, telencephalon, lobe, parietal, area, primary somatosensory cortex, function, perceive contralateral touch. But notice where this arrow is, I could also ask you to identify the structure, and the structure is the postcentral gyrus. So those are the types of questions you will be asked on the cerebrum here. Good news is, for every single one of these, the answer to the cephalon is the same. Everything we've looked at so far is the telencephalon, and of course, if you go back to page 1, you see on page 1, this is all under telencephalon, because that's the cephalon that this is a part of. We've divided it up into the 4 lobes, and we've identified the areas and their functions and some structures as well. All right. Now, not too subtly, I have mentioned that there are 4.5 lobes, because there are some individuals that believe that there is a 5th lobe associated with the cerebrum. And that 5th lobe happens to be nestled right inside of our lateral sulcus. So I think what we need to do now is take another break, because we've been going for a bit here. So we will take our next break at this point, take a quick 15-minute break, and come back and talk about this potential 5th lobe. All right, so let's see. It looks like it is 10:35. Am I seeing that right? 10:35. So let's restart at 10:50. And at that point, we will start the recording again. All right, any questions before we take a 15-minute break? All righty, see you in 15 minutes. All righty, so we left off and we were getting ready to dive into the lateral sulcus. As I mentioned, here we see this great picture from your textbook where it's kind of opened the space up. But I think where we see it best is if we go back to this coronal section we were looking at before. Notice if you follow, and I'll just use white the lateral sulcus in, there is this big fold of cerebral cortex in this space. This big fold of cerebral cortex in this space is a structure known as the insula. And the insula is a very large cortical structure, so there are some individuals who kind of refer to it as a fifth lobe. Whether you call it a fifth lobe or not, I don't care. I'm not going to ask you how many lobes are in the cerebrum, but we do need to know what the insula is. The insula basically is the cortex that coordinates the activity of all of these nuclei that we see in here, and we'll talk about these in just a minute. So this region is what is known as the it's what's known as the limbic system. Now, I've written this definition down because I want to read it precisely to you so that it makes some sense. So what I have here, and I wrote this down, so again, I want to read it to you, and I'll read it to you slowly. The limbic system is these cluster of nuclei. So again, this is the gray matter that is basically between the cerebral cortex and the diencephalon. And essentially, it is a functional group. And it functions as the emotions, memory storage, and retrieval. But here's the part that I wrote down that I wanted to say. It links conscious thought and unconscious function. What do I mean by that? So again, this is involved in emotions. I'll put it all out. Memory, storage, and retrieval. But also this linking conscious thought and unconscious function. What do I mean by that? Linking conscious thought and unconscious function. Anyone have any idea what I mean by that? All right, let me ask the question a different way. Back in ancient times, we used to be able to go out. So on a Wednesday night, you decide to go out to a business meeting with some of your colleagues, and you come stumbling home, rip-roaring drunk at 3 o'clock in the morning on a Wednesday night, fumbling with your keys, making a tremendous amount of noise, incapable of getting into your house. And your significant other comes up and unlocks the door to let you in. And without them even saying a word, can you tell what their attitude is towards you at that period of time? The flushed face, that blood vessel that's pulsing on their forehead, all of those types of things? Absolutely. Does what you think, your conscious thought, affect your body physiologically? When you're scared, when you're angry, when you're happy, when you're horny, when those types of things occur, are there physiological changes that take place in your body? Absolutely. And that is what this limbic system does, right? If you think about it, it's kind of the whole concept behind a lie detector test, right? The police officer hooks you up to all these machines that measure your blood pressure, measure how much sweat you're producing by the conductivity of your skin, right? Your heart rate, all of these different types of things. And they ask you all these benign questions. What is your name? What is your favorite color? What was the puppy's name, your first dog's name when you were growing up? Where were you on the night of the 5th, right? Because they want to know when you're having that emotional response to something that you're going to lie or something bad happened, right? Our body changes physiologically because of that. And that is what this limbic system does. This limbic system links what we're thinking about with physiological changes in our body. All right? And it starts with the insula. The insula, that little bit of cortex, and again, I'll highlight it here in red just to emphasize this. This insula over here, as I mentioned, is the cortex associated with the limbic system. It plays a role in pain, it plays a role in desire, it plays a role in anger, but probably one of the most interesting functions associated with this insula is addiction. Right? One of the things that they have found is people that that partake in addictive behavior tend to see an increased activation in an insula. Right? You can take 10 different people, put them in, again, we were talking about one of those functional magnetic resonance imaging machines, and have them all smoke their very first cigarette. And what you'll see is obviously smoking that cigarette is going to affect all of their bodies, however, all of their brains. In some of them, it will light up that insula. The insula will become active with that activity. And those people whose insulas are activated by that activity are more likely to be addicted by that behavior. And it isn't just chemical things or drugs or things along those lines. Gambling is probably a great example, right? Something as simple as guessing a coin flip, heads or tails. You can put people in magnetic resonance imaging, flip a coin and ask them to say heads or tails, and they'll do that. And people who are addicted to gambling will see increased activity in that insula, whereas people who are not addictive to gambling do not see that same activation. This is powerful information because we now know that if there are ways that we could modify the behavior of that insula, it might be a way to break some of these addictive behaviors more successfully. So again, it's a really interesting field of study where there is a lot of work going on in there. But notice there's a lot of other stuff in this limbic system as well. Here we see one view of them. Let me go ahead and clear this for a second. It's a little tricky to see. I think this picture does one of the better jobs of showing where it is. Notice we are seeing a partial sagittal section. They've removed the frontal and parietal lobe, but notice they've left the temporal lobe in place because these are midline structures, and these also are things that are out here laterally in the temporal lobe. So again, we see that from this view, deep here, deep to the parietal, down here in the temporal lobe. Let's talk first about this one, this purple one. This purple one, we see it there and we see it here near the front of the temporal lobe, is an almond-shaped structure, which is where it gets its name, known as the amygdala body, or the amygdaloid body, or the amygdala. The amygdala plays an important role in recognition of emotions. In particular, fear and anger. Your amygdala is what we in my family commonly refer to as the worry monster. Right? The worry monster is that survival tool that we have from an evolutionary standpoint. Again, evolutionarily, we all lived in trees. Evolutionarily, bears with axes roamed the countryside. So any sound, any snap of a twig, something along those lines could be a bear with an axe that is threatening to kill us. All right. Now we live in gated communities and locked houses on the second floors, locked in our rooms, but that worry monster is still there. So you fear the monster under the bed, or that, bad people are going to break into your house and steal you away. Right? That amygdala is your worry monster. Its job is to keep you alive, and so it over-exaggerates those fears for us. Another way we see its function is in monkeys. Monkeys are excellent at nonverbal communication. They have very distinctive facial expressions that they will say. Some of them are submissive and some of them are aggressive. And so what they'll do is they will put a monkey in a chair, kind of like I'm sitting here in the chair. And basically they have two buttons to push. And what happens is they will show them an image. And if it is an aggressive monkey face, they'll push the aggressive button. If it's a submissive monkey face, they'll push the submissive button. And when they do this activity, they get rewarded with juice. They like juice a lot, so they'll sit there for hours and do this activity. And they're excellent at recognizing the submissive faces and the aggressive faces, unless there's a problem with their amygdala. If there's a problem in their amygdala, they're not able to differentiate those faces anymore. And basically they can't tell which ones are aggressive and which ones are submissive. So that amygdala is that fear and emotion. It is that worry monster that we have. All right. Questions on that one? All right, let's talk and let's switch gears to this picture to take a look at this red structure, this lateral structure that is out here. Now this lateral structure, the picture does a little bit of a disservice to us because what it actually looks like is it comes around and then it has kind of a larger belly to it. And then the really big part at the end is really this curled up region of the cortex. So what's really in here is an enlarged belly and this curled tail. I make a point of emphasizing this because that's where it gets its name. If you notice, it looks a little bit like a seahorse, and that's what hippocampus means. Hippocampus means seahorse. The function of the hippocampus is to convert long-term memory into, short-term memory into long-term memory. All right, I'm going to give you 3 objects: ball, boat, car. Somebody say them back to me.
Ball, boat, car.
Car.
Excellent, right? Many of you were able to do that, and you were able to remember those 3 things, right? And anybody can do that. However, someone who has damage to their hippocampus can still remember those 3 objects. And what were those 3 objects again?
Ball, boat, car.
Ball, boat, car. They can keep that because it's in their working memory. The problem is you then give them you give them a sheet of mathematical equations. Doesn't have to be hard stuff. Stuff like 7 minus 4. What's 7 minus 4?
3.
2 plus 7.
9.
4 minus 3.
1.
After doing a sheet of those, you can ask them for those 3 objects. And what were those 3 objects again?
Ball, boat, car.
Right. A person who has damage to their hippocampus can't do that. They're not able to convert their short-term memory into long-term memory. They lack that ability. They have working memory, but once that information's out of their working memory because they're doing those mathematical problems, they lose that piece of information. There is probably one of the most famous individuals that have dealt with this condition is an individual who's known just by the code HM. Patient HM, gosh, 35 years ago now. He might not even be alive anymore. Wow. When I was in grad school, it was just coming up on the 25th anniversary of his accident. He was camping, and as he was out camping, it started to rain. And so he had taken his barbecue and put it in the car with him while he slept in the car. As a result of that, he ended up getting carbon monoxide poisoning. And the carbon monoxide poisoning basically deprived both of his hippocampi of oxygen, and he lost the ability to convert short-term memory into long-term memory. One of the instructors who was at UC Davis at the time while I was there was one of the first neuroscientists to work with him and had worked with him for close to 25 years. Like I said, it was going on the 25th anniversary of them working together, researching with this individual during that time. And in those entire 25 years, every morning when he walked in to work with that individual, he had to introduce himself to him again. When he left to go get lunch and came back in the afternoon, guess what he had to do? Introduce himself to the person again. Right? The person was not able to remember that researcher that he'd been working with for 25 years because he lost that memory. He lost that ability to convert short-term memory into long-term memory. He could remember everything that happened up to his accident, well as any one of us can remember things. I mean, how many of you can remember what happened 25 years ago? Hell, most of you aren't 25 years old, right? So obviously that's challenging. But again, when it happened up to the point of the accident, he had a pretty normal memory, which you would expect to be a normal memory of being able to remember things up to there. He just hasn't been able to produce any new memories. Now, obviously, when you wake up in the morning and look at yourself in the mirror, if you haven't remembered the 25 past years, right, the person looking back at you in the mirror is not the person you expect to see. Exactly. Yep, absolutely. We'll talk about that in just a second. You are absolutely correct. Absolutely. So he is aware that something happened and time has passed. His amygdala was also damaged by this, so his emotions are also somewhat muted as well. So he doesn't freak out quite as much as you might expect him to when he first looks at himself in the mirror and sees somebody that he doesn't know. But we've also learned a lot about different types of memory. We understand about working memory, as I mentioned, versus the long-term memory, but also muscle memory. Not surprisingly, this individual's got a tremendous amount of time on his hands. And so one of the primary things he does is play video games. So what they will do is you can walk in, and of course you have to introduce yourself to him, and then you'll show him a video game. Have you ever played this video game before? And he'll say, no, I've never played that video game before. You then put it in the machine, give him the remote control, and he'll get the top score on it. And the entire time he's playing it and crushing the game, he will swear he's never played the game before in his entire life. So that muscle memory, right, that motor memory of playing that game is something that he's obviously retaining even though he's not aware of it, right? Again, think of the implications of this. You could teach him to play the piano, and each day he would get better, but he wouldn't be aware of it. So if you were to theoretically do this for 2 years, you could come in and say, hey, do you know how to play the piano? No. Well, just give it a try. And you give him the piano in front of him, and he would play a beautiful song and have no idea how he knew how to do that. Now, Madison is correct. There's not one, but two equally amazing movies, dramatically different movies, but equally amazing movies that involve people that have hippocampal damage. The first one is the comedy, 50 First Dates, a pretty good movie and a pretty good representation of someone who has hippocampal damage. Anyone know what the second hippocampal damage movie is? Also an equally amazing movie, but it is a drama, not a comedy. It's been a while, so it's not quite as popular.
Would it be Shutter Island, maybe? But that isn't it.
I'm not familiar with that one. I don't know that movie.
It's a Leonardo DiCaprio movie.
Oh, is that a memory one? I haven't seen that one. No, the one I was thinking of is Memento. Again, Memento's
that's a
little bit older, but Guy Pearce is a policeman who a bad guy breaks into his house, kills his wife, injures him, and he gets this hippocampal damage. So he can't convert short-term memory into long-term memory, but being a policeman, he wants to revenge the death of his wife. So he's constantly taking notes and putting tattoos on his bodies and taking pictures and stuff like that. And if you watch the movie, if you get the DVD, back in ancient times, movies were on DVDs, there was a special code you could put in, because when you watch the movie, you don't watch it synchronously in time. You get the breaking up of the time because he doesn't see things synchronously. And there's a code you can put into DVD where you can actually watch it in chronological order. So you can watch the events in chronological order. Whereas the movie breaks things up. So again, an equally amazing and fascinating and interesting movie, but again, all of it involving this concept of being able to convert short-term memory into long-term memory. All right, questions on that? All right, let's look at another structure that we have in here, one we have talked about before. Down here, underneath the cerebral cortex, we see an enlargement and a long stalk-like structure that comes off of it, the olfactory bulb and the olfactory tract. Anyone remember what the olfactory bulb and olfactory tract are involved in? Sense of smell. Excellent. Remember, we talked about it, and let's go back to, did I close that? No, here, this is where we got it. So hold on, I need to close that. No, whoops, wrong button.
You have to have a time, don't do it.
Hold on. There we go. Here we go. So where did it go? Perfect. Remember, way back in ancient times, we were talking about the bones. And when we were looking at the sagittal section of the brain, one of the things we talked about was that cribriform plate that sits right there, and that it has the olfactory foramina in them. And if we look closely, and I will narrow the size of my pointer for this, here is that enlarged bulb, the olfactory bulb, that sits on the cribriform plate, which, as we talked about, is where the olfactory nerve comes through those olfactory foramina, giving us that sense of smell. And I told you the story about how my sister's boyfriend fell down the flight of stairs and ripped that olfactory bulb off his cribriform plate and lost his sense of smell. Well, this olfactory bulb is where the cell bodies are located, and the axons come off, forming a structure called the olfactory tract. And if we peel away that pesky skull, that is what we see here. Here we see that olfactory bulb and the olfactory tract and how it goes straight to the limbic system. Our sense of smell is special in that it is the only sense that goes directly to our limbic system. Remember, our limbic system is the one associated with emotions and memory. So if, like me, you're longing for that long lost boy that you broke up with, so you have those pictures of him that you look at that make you sad and dreary, or maybe you listen to that last recording he left on your phone that you haven't deleted yet so you could listen to him. But when you're really feeling melancholy and really miss him, you do what I do. You grab that t-shirt of his that you have out in that Ziploc bag and you open it up and just get that little bit of smell of him. And that little bit of smell brings all the emotions and all the memories back. Our sense of smell is the one that is closely associated with emotion and memory. Or maybe it's the smell of fresh baked bread or that apple pie that grandma used to make that reminds you of the summers you spent at grandma's house, swinging on the tire swing, swimming in the pool, right? All those kind of things. Our sense of smell is most closely associated with a memory and motion because it's the only sense that goes directly to our limbic system. Along that line is a structure, this one right down here, and of course it's red and I'm coloring it red, so that makes it a little trickier to see. We see it here as well. These are paired structures on both sides of our cerebrum. So if we look at it from an underneath view, and I think we have to go a little bit of a way to get to it because there we go. Notice, and again, remember, you have to remember, anatomists are sad, lonely individuals. So we see these two round bulbous structures sitting side by side. So if you're a depraved anatomist, what would you call them? They are the mammillary bodies, right? Because they look like little boobies. All right, so we have these mammillary bodies. Where's my picture? This is the one I want. This mammillary body is job, if you look at it, it basically, I'll use a line for this one. Notice that there are fibers from our sense of smell that come to it. And then there are also fibers that come up and connect it to the hippocampus. The reason for this is our mammillary body is basically what links our sense of smell to our memory. In particular, for smell and taste aversion. Right? Your job is to stay alive. So if you happen to be a sheep out in the field and you smell some little red berries and you eat those red berries and you get sick, do you want to eat those red berries again? No. So we have this sense of smell and taste aversion, right? Or maybe you don't even have to be a sheep. Maybe you're my wife. When my wife was pregnant with our first child, We were down in Southern California. There was a restaurant there that she really, really enjoyed. And what she loved about that restaurant is they had this peppercorn ranch dressing that they used on the salad that she really, really loved. So we would eat it a lot when she was pregnant. However, one night after eating it, because she was pregnant, got sick and threw up a whole lot. Was it because of the peppercorn ranch? No, it was because she was pregnant. However, having thrown up what felt like gallons of peppercorn ranch, did she ever want to eat that peppercorn ranch ever again? No. Or maybe you're in high school trying to finish off that gallon jug of Jose Gold tequila, right? And you ran out of lemons as chasers, and try using oranges. And to this day, the smell of fresh squeezed orange juice still makes you a little nauseous, right? That ability to link something that we smell or taste to something that made us sick, so we want to avoid it again, that is what the mammillary body does. Now, I'm a little confused. I've spent all this time talking about the mammillary body, but I don't see it here in the limbic system. Does that mean I've wasted all this time because you're not responsible for it? Yes, Dr. Sutsky, you love wasting our time. And that is true, but not in this case. No. Is your mammillary body on your list?
Yeah, it's in the diencephalic features.
It is a diencephalic feature. So it is something we're responsible for. Not in this part of the brain, but it is something we're responsible for. But that is why we're talking about it here. Because notice this yellow structure, and I'm going to go ahead and use my purple highlighter to emphasize this. This yellow structure is a bundle of axons that connects the mammillary body to the hippocampus. And this yellow bundle of axons is a structure you are responsible for on the exam. It is a structure known as the fornix. Notice this picture is nice because it shows it completely intact, so we see the entire thing. But notice also we see it in a sagittal section as well. Notice they've been nice enough to color code it yellow for us to see it there. But even if we go to the half head chart, we don't see the hippocampus. We don't see, actually that's not true. We do see the mammillary body. Let's color it red because that was the color we've seen. This here is the mammillary body on our half head chart. But what we're interested in, and I'll use yellow, is this structure here. This structure here is that fornix. So the fornix is one of the structures on your list you are responsible for. It's just a bundle of axons. It carries action potentials from one location to another. So it doesn't have a true function other than being a bundle of axons, but it is one of the bundle of axons we need to be able to identify in our limbic system. Notice it's white on our chart here. That makes it easy to be able to recognize. But notice it's not the only white structure. In fact, the biggest white structure is this big comma-shaped structure right here. This big comma-shaped structure right here is a big, huge bundle of axons known as the corpus callosum. This corpus callosum is the largest commissural tract. Remember, commissural means that it's axons going from one side to another. We see it very, very nicely on the chart here, but the other place we see it really, really nicely is not just here with this view, where we see it right here, but notice if we change our view to this view again. Notice here, these axons going from one hemisphere to the other hemisphere, just below the longitudinal fissure is the corpus callosum. So the corpus callosum is the biggest bundle of axons going from one hemisphere to another. Makes it easy because it's always going to be white matter. It's always going to be those axons. Easy to see and to identify. Obviously, it is important to be able to coordinate what happens, right? At the end of class, as you guys know, of course, you guys are also thrilled and with the fact that I have provided you with all this knowledge, that of course you guys always stand up and give me that standing ovation at the end of every lecture. Well, obviously, if you're going to give a standing, can't even think of the term now, ovation, a standing ovation, you have to be able to coordinate the activity of your left hand and your right hand so you can bring them together. And so obviously fiber tracks connecting the two sides together should play a really important role in that, right? If someone cut their commissural unit, if we were to have somebody who, for some unknown reason, had their corpus callosum cut in a condition we call a split brain, they wouldn't be able to function hardly at all, would they? Would splitting your brain like that by cutting your corpus callosum dramatically affect your behavior? What do you think? Yeah, you definitely would think that it would, wouldn't it? Turns out it doesn't. We think it would, but shockingly, people with split brains actually function pretty normally. In fact, it's very challenging in some cases to tell a person that has a split brain. There are some special tests that they can do, but it's some very unique tests. One of them is they look in this viewfinder, and as they look in the viewfinder, what they see is they see an image of an individual. One side it is male and one side it is female. So what they'll ask them to do is they will ask them to write down what they see, and they'll ask them to say what they see. And what'll happen is they'll write down male and they'll say female, and they won't be aware of the discrepancy. But in the real world, is the half of the right side of someone male and the left side of someone female? No, in the real world, there's consistency, and that consistency allows these people to survive and actually thrive pretty normally. Of course, the question is, why would you even want a split brain? Right? It seems like an odd place to put a piercing or something along those lines, so it doesn't seem like it would be something that would commonly be done. Anyone know why someone might get that split brain procedure done to them? There you go, epilepsy treatment. Epilepsy is a condition where basically what happens, a region of the cortex produces elaborate and erratic action potentials. And those erratic, elaborate action potentials spread over the cerebral cortex in a process that is called a neural storm. And this neural storm can spread across the entire cerebrum and can cross the cerebral, the corpus callosum to go from one side to the other. In extreme cases, these can become so powerful that these people have major epileptic seizures where they're flailing on the floor, worrying about them biting off their tongue as a result of that, right? They obviously can't drive a car. It can be very challenging for them to lead normal lives. Now, the one good news about it is often when those epileptic neural storms occur, they typically always initiate on just one hemisphere of the brain. So if what they're able to do is to go through and split the brain by cutting the corpus callosum, that limits the spread of that neural storm, and it can dramatically decrease the symptoms associated with them. Madison, you said your mom had epilepsy. Did she have this procedure done, or was she able to treat it with medications? Because some medications are able to decrease the amount of neural storms that people have. But in the cases where the medicine doesn't work, this split brain procedure is something that they sometimes consider doing.
So she never got the split brain procedure. She still has it. I'm the one who had it. I grew out of it as a kid, but she still has it. She does medications, and I think it's like, what, once a year you go in and you get twice a year. She's standing right next to me eating. She goes in, see if they need to reevaluate her medications. She did have a seizure last year in October or so.
Wow.
And it, wasn't allowed to drive for 30 days. And it was like the first seizure that she had in like 10 years. So
That's good. Well, so it sounds like that she's, again, the splitting the brain is a very radical procedure when it is done. But like I said,
I went to high school with a girl who got that procedure.
Yeah, as debilitating as these seizures can be, it is, while it is dramatic, it can actually cause significant improvement for some individuals. So absolutely, so it is something. And again, surprisingly, it's not nearly as debilitating as one would think to have, because again, there are other commissural tracts, so the two sides are able to still coordinate, communicate with each other, but not nearly as dramatically as obviously when you cut that big, massive corpus callosum. All right. Excellent. Thank you for sharing that personal information. We really appreciate that. All right. I want to point, while we're here on this picture, one other thing, actually two other things to you. So again, let's go ahead and draw the corpus callosum again. So here is that corpus callosum. The other thing that I want to point out to you that we see on this illustration is that, and I'll use blue for this, our nervous tissue is not completely solid. There are some fluid-filled spaces inside of them. Here we see a couple pieces of a fluid-filled space known as the lateral ventricles. Notice if we switch views, I'm going to come back to this picture, to, where was the picture? I thought I had a different, better picture than this. There it is. Excellent. Notice here we see this big C shape. Oh, I like this view. Let's take a look at this one. This big C-shape space that is filled with cerebral spinal fluid. Notice here is that corpus callosum. It has been cut. And notice right here, separating the two lateral ventricles from each other are these thin membranes. Notice here is the fornix. Here is the corpus callosum. And basically between them is this thin membrane that separates the two ventricles from each other. This membrane is what is known as the septum pellucidum. So here from this view, we see it really, really nicely, really clearly separating. Notice also, and again, slightly different in artistic license here, notice here, we see that septum pellucidum as well. Here is that septum pellucidum that is separating the two lateral ventricles. But the other place we see it is here in the sagittal section. Notice again, here is the corpus callosum. Here is the fornix. And remember, as we said, in between is this membrane. So this membrane right here, in between the corpus callosum and the fornix, is that septum pellucidum. So if this was a real brain and we were to poke through there, we would be in that space that is the lateral ventricle. So that is like the fornix. These, like all three of these really are just structures. These are structures that you need to be able to identify in a sagittal section or in some other view as well. All right. One structure left we have to identify in the limbic system. Oh, hey, look at that. Pretty picture. So for that, let's go back to here. The last structure you need to be able to identify on the limbic system is actually a collection of nuclei. These three nuclei together, notice one of them is the putamen, one of them is the globus pallidus, the other is the caudate nucleus. As I look at my list, are any of the three of those names you need to know? Do you see them on your list? No. So remember, like with the muscle group, you just had to know levator ani. You didn't have to know iliococcygeus and pubococcygeus. It's kind of the same thing here. You just need to know the group name for this. The common group name for this is the basal ganglia. Now, of course, oops, helps if I spell it right. Basal ganglia is the common name. It is what most commonly referred to. However, remember, a ganglia is a cluster of cell bodies outside the central nervous system, and this is in a peripheral nervous system. So anatomists have been using this name basal ganglia for damn near ever to talk about them, and they've been completely misnamed. So the good news is they are getting a little better, and more people are referring to them as the ganglia, as the basal nuclei. But because the term basal ganglia is so ubiquitous out there, I will allow you to use either of those terms. So these three things collectively are what are known as your basal nuclei, or you may call them the basal ganglia. So then the only other thing we need to do is know what they do. They function basically as your autopilot. Now, what do I mean when I say autopilot? What's an autopilot? Not a trick question. What's an autopilot?
Doing something without thinking.
True. Where do you find autopilots? Where does the term autopilot come from?
The plane.
Plane, absolutely. Does the autopilot take off the plane? Does
No.
the autopilot land the plane?
No.
No. But once you get it up to the height, the elevation that you want, the autopilot will maintain the flight. And that's what happens here. Your autopilot, your basal ganglia, maintain a learned behavior. It doesn't initiate the behavior. It doesn't terminate the behavior, but it maintains a learned behavior. Walking is probably one of the greatest examples of this. I know a couple of you have young kids at home, right? Either trying to learn to walk or have recently learned to walk. When that 1-year-old is trying to learn how to walk, it requires all 110% of their concentration. They're thinking about what every arm and what every foot and what every toe is doing as they're trying to make those movements and learn how to walk. Does walking require all 110% of your attention anymore? No, absolutely not. You're eating a sandwich and tweeting and TikToking and doing all these things while you're walking, not talking to your neighbor and doing all these other things without having to pay any attention to it anymore, right? When you first started driving, it required 100% of your attention as you're doing it. And again, now you're eating and TikTok dancing and tweeting and eating a sandwich and doing your makeup and doing all these other things while you're driving. It maintains that learned behavior. Of course, there is a problem with that because you get in your car and your autopilot kicks in and suddenly you're halfway to work and you were going to the store to get milk, right? Or you're in the shower and your mind starts wandering and you can't remember whether you washed your hair or not because your autopilot kicked in and you went through whatever your automatic sequence is and you weren't thinking about it. Or my favorite example is knitting, right? My grandmother tried to teach my wife how to knit. And it required every single ounce of attention of my wife to try to figure out the knit and the purl and all of those things. Does grandma have to spend her attention like that? No, she's watching TV and having a conversation and baking a pie, all while she's whipping at that stocking at record speed, right? That's because we have this autopilot. It can maintain a learned behavior. We have a learned behavior, it can maintain it without requiring our attention. And that's basically what our basal ganglia does for us. All right. Questions on that? All right. Excellent. With that, we are done with all of the structures we are responsible for in our telencephalon. We still have some spaces we need to talk about. We've already talked a couple about them, like the lateral ventricles, and those spaces are going to contain our cerebrospinal fluid. But I think it'll be better to wait till the end to talk about that. So what I want to do is jump ahead to the diencephalon and talk about the diencephalon. So any questions on the telencephalon before we move down to the diencephalon? All right, excellent. Let's move to the diencephalon. That one, that one, that one. There, perfect, excellent. So the diencephalon, this picture doesn't do a tremendous amount of service. Here we see the representation of it deep beneath the cerebrum. And again, notice in that illustration, even when they peel it off, we get a little bit more detail, but not a whole heck of a lot. However, one of the places where I think we see the best example of what it looks like is in, there we go, our sagittal section. Here we see it in our illustration, and then here we see it in our chart. And let me go ahead and outline the area. Basically, this area right here that area right there kind of looks a little bit like the duck's head, is the diencephalon. Notice if we go back to the illustration, we see that same thing. So again, this area here, there, around there, that area there is our diencephalon. All right. Now, the most prominent structure, well, one of the most prominent structures we see in the diencephalon is this kind of eye-looking structure of the duck head. It is a big, round, globular structure that is known as the thalamus. Here, we see a picture of what the thalamus looks like, and the thalamus is actually a collection of nuclei. Right, collection of gray matter located basically at the top of our brainstem. And its job is basically to be the relay center. Almost all information that comes into your brain has to pass through the thalamus first. Notice there's that pesky word, almost all. We've already seen how our sense of smell goes straight to the limbic system. But every other sense and most of the other information that comes into our brain comes through here. So we have all these different nuclei. Notice we have a big nuclei for a cerebellum where our hearing comes in. And notice this tiny little bundle right here is a structure known as the lateral geniculate nucleus. This is where your visual information comes in. Now, you don't technically need to know that, but that happened to be where I did most of my research. And my research grant actually requires me to say the term lateral geniculate nucleus at least 3 times in class. So now we've done 2. So there you go. So that was the area where I studied in the brain, that tiny little nobule where a cluster of cells are located, where our visual information comes in there before it gets sent to the occipital lobe to be processed. So this is our relay station. Sensory information, hearing information, all of these things come into the thalamus first before they get sent to the cerebrum. And they're paired. You have 2 of them. And they don't touch. There is a little space in between. That space in between is your third ventricle, where your third ventricle is located. However, there is something interesting. They don't touch. What do I do with my pencil? I don't want to use a pen. There's a pencil, that works. They don't touch the 2 thalami. You have 2 thalami, they do not touch. However, is it important that information from one thalamus be able to coordinate with the information of the other? Yes. So there is a small bundle of axons that connect them together. Notice then, if you were to cut the brain in half, when you cut the brain in half, what would happen as you looked at it from the side, not only would you see the thalamus, but you'd see that little bundle of axons that sticks out as well. And that is exactly what is happening in this image. In this image here, and I'll use my highlighter first. This is like looking into a bowl. If you look into the bowl, you see the sides of the bowl, but you know there is a space in there. And that's what we have here. In here, this whole thing is a space known as the third ventricle. And the thalamus helps to form part of the wall of that. So this space is the third ventricle, but this structure right here is the thalamus. And, change colors, this one little white fiber that we see there, the eye, the pupil of our eye, is that point that sticks out where the fibers connect. And any idea what that bundle of axons might be called? Only had a list of terms in front of me that I was responsible for. I might be able to figure out what that bundle of axons that connects the two thalami together might be. Because maybe it's this next thing on the list that I'm responsible for knowing. There you go. Excellent. The intermediate mass. So what I've colored red is your intermediate mass, and what I have colored blue is your thalamus. All right.
So the intermediate mass joins the two?
Yep, joins the two thalami together. So the
Okay.
two thalami are connected by the intermediate mass. Yep. All right. With me so far? Excellent. Now we are still looking in our bowl. So notice what looks here like the smooth wall of our bowl is a collection of structures, and these collection of structures are below the thalamus. So what do you think the collection of structures below the thalamus might be? The hypothalamus, absolutely, excellent. Now here, the picture makes it look like just smooth tissue. However, if we look at one of the different pictures from your textbook, this one right here, we see that under the thalamus there, again, it is a collection of nuclei. Like the thalamus, you don't need to know the names of the individual nuclei, but these individual nuclei serve all sorts of important functions. Your hypothalamus is one of the main regulatory structures of your body, right? Let's stay at this view. I like this view for a second. We'll come back to the other one in a moment. Notice underneath here is a structure called the pituitary gland. The pituitary gland is the master endocrine gland. It produces over a dozen different hormones. And many of those hormones are things like the thyroid stimulating hormone. Guess what thyroid stimulating hormone does? Not your question. What do you think thyroid stimulating hormone does? There you go. It stimulates the thyroid gland. Exactly. So the pituitary gland doesn't only produce dozen or more hormones, but many of those hormones control the other glands of the endocrine system. So it's called the master endocrine gland. And for the longest time, it was believed the pituitary gland was the command center for our endocrine system. But only about 25 years ago, which you think about it in the grand scheme of things is not a very long time ago, we actually learned that it is actually the hypothalamus that controls the pituitary gland. Yes, the pituitary gland controls all the other endocrine glands, but the hypothalamus controls the pituitary gland. It's like in politics, right? The president is the figurehead, like the pituitary gland. The one in front, the one that seems to be in charge, right? The one that seems to hold all the power. But we all know that behind the scenes, the Illuminati truly controls everything in the world, right? So behind the scenes, there's the Illuminati, and then you have the figurehead in front. And that's what we have here. The hypothalamus is the control behind the scenes. The pituitary gland appears to have all the power, but it is actually controlled by the hypothalamus. All right? So, the hypothalamus is a major regulatory organ. It plays a role in regulating heart rate, body temperature, things along those lines. And it controls, it is the command center for our endocrine system. Now, notice also, if it is going to control the pituitary gland, the two have to connect to each other. So notice there is a stalk-like structure that sticks out of the bottom of the hypothalamus that connects the two together, and that structure is the infundibulum. Notice our illustration emphasizes that stalk a little bit better here. But the other place we see this, and you may remember it when we talked about it, notice, let's come back here. Let's use blue. Let's use drawing. Here again is the hypothalamus, this area here. Here is the infundibulum, and notice right here is the pituitary gland. Notice that pituitary gland sits in this U-shaped bone feature. Anybody remember what that U-shaped bone feature was called? Sela turcica, because it has a Turkish saddle shape to it. Although there was another name. Anybody remember the other name we used for it? It was the hypophyseal fossa. Fossa is an indentation. Hypophyseal, hypophysis, is the fancy word for pituitary gland. So it is the indentation for the pituitary gland. Hypophyseal fossa means indentation for the pituitary gland. And so that pituitary gland sits right there in the sela turcica, underneath the hypothalamus. All right. Questions on that? All right, let's take a look at one other structure. Here we see this nicely. Perfect. Notice those are those mammary bodies we already talked about. Here is the infundibulum, and then of course, as we know, the picture doesn't draw it, but the pituitary gland would sit right there on the infundibulum. This is an inferior view or an anterior view that we were looking at it. So they've moved it out of the way, and the reason they've moved it out of the way is so we can see this white matter structure underneath it. This white matter structure underneath of it are axons coming from the eyeball. Starting first with the optic nerves, which happens to be cranial nerve 2, one of the cranial nerves we will be responsible for there. But notice they come together, right? One of the things we've learned is that if I want to move my right hand, I make that decision on the left side of my brain. If I want to feel something with my right hand, I process it with the left side of my brain. And if I want to see something on the right side of my body, the visual field, I process that with the left side of my brain. So what happens is information from the eyes, some of it has to cross. And it crosses at this specialized structure right here, known as the optic chiasm. This optic chiasm is that special crossing point. Once it leaves there, they then form the optic tracts, which carry the information to the lateral geniculate nucleus. And then that goes from there, from the thalamus to our occipital lobe. Let's take a look at a pretty picture that shows how this works. Here we go. So notice, here is my left eye. This is a top-down view. If you think about the way information comes into my eye, some of the information that comes into my eye comes in from the right side of the world, and we want that information to stay on the left side. So those axons stay on the left. But other information comes into my eye from the left side of the world, and I want that information to cross. So notice here in the optic chiasm, we have a partial crossing of information, so that everything from the right side of the world from both eyes goes to the left side of my brain, and everything from the left side of the world goes to the right side of my brain. We'll talk about this more if we get to the sensory structures, but for now, all we need to know is the name of these three things. The first part of this is the optic nerve. The second part of this is the optic chiasm, and the third part of this is the optic tract. So optic nerve is before it crosses, optic tract is after it crosses, and the optic chiasm is where it crosses. All right? Questions on that? I do want to show you one more interesting thing. Notice here, the optic chiasm is right above the pituitary gland. And in fact, actually, let's go, I think this view shows it nicely. So let's go ahead and draw my circle again. Here is the, oops, pituitary gland right here. And notice, in red, I'll make a red block, this right here is the optic chiasm. That's where the information crosses. This relationship is significant because one of the things that can happen to individuals if they have a pituitary gland problem is their pituitary gland can swell and enlarge if you have a pituitary gland disorder. Notice it's stuck there in the bone, so it can't go down, it can't go forward, it can't go back. It can only grow up. And as it enlarges and grows up, what happens is it pushes on the optic chiasm. So there you are sitting at home, and suddenly you get this tunnel vision. You can't see anything from the periphery anymore. All you can see is tunnel vision right in the front. You go running to your optometrist, and can your optometrist do anything for you in this case? No, they're practically useful individuals anyway, but in particular, in this case, they can't do anything because the problem is the pituitary gland. So instead, they shoo you off to an endocrinologist who is going to help you with that condition. But so then notice, by pushing on that nerve, we get this visual deficit where it can actually cause a tunneling of vision by squeezing that optic chiasm, pinching those nerves that are carrying the crossing information. All right. While we're here, we can see two other things as well. One of the things that I've tried to emphasize is that the corpus callosum is our largest commissural tract, but it isn't the only one. So let's draw them. I like this. So here we'll highlight it in purple. Here is our corpus callosum, one of our commissural tracts. Here is our intermediate mass. It crosses the two hemispheres as well. But there are two other bundles of axons that cross from one hemisphere to the other. One of them, which I'll draw in red, is this one right here. And the other one that I will draw in blue is right here. Notice one of these bundles of axons that goes between the hemispheres is towards the front of the brain. One of them is towards the back of the brain. So what do you think we call this red bundle of axons that goes between the hemispheres? Only I had a list of the terms I was responsible for. Anterior what? Anterior commissure. Excellent. And that makes the blue thing? Posterior commissure. Excellent. Notice they're pretty easy to find. The corpus callosum kind of shows you where both of them are. We can go from the front, we can go from the back. Although really, if you think about it, the fornix points right at the anterior commissure, and then the posterior commissure is right behind it. The problem with following the corpus callosum back is there's a bump in between. So let's make that bump in between orange. This bump in between right here is what is known as the pineal gland. Anyone know what the pineal gland or the pineal body does for us? It's your third eye. It's your first chakra. But what does it actually do for you? Here, I'm going to cheat and give myself a little writing room. Come on, I know somebody knows. What does that pineal gland do for us? Go ahead. It produces what? Oh, wait, look it up on your phone if you have to. What does the pineal gland do? But it doesn't just regulate any hormone. It produces and releases a specific hormone. What specific hormone does your pineal gland produce and release? There it is. Excellent. Excellent. Melatonin. That's that thing that makes our skin dark. Excellent. So it's our pineal gland that makes our skin dark. All right. Questions on that? Really? You guys are just going to let that go? There you go. Melatonin isn't the thing that makes your skin dark. What makes your skin dark again? Melanin. Excellent. Melatonin, absolutely, as Adina pointed out, plays an important role in our sleep-wake cycle. Now, you do indeed have a sleep-wake cycle. I know most of us don't get to actively participate in it. Right? I'm guessing there are a few of you this morning who woke up when you wanted to. Right? Either your alarm went off or your dog woke you up at 4 o'clock in the morning because you needed to go outside, or any number of other things. Your kids came bumping into the room, right? Typically, when we wake up, when we go to bed, when we eat our meals, all of those are bound by social constraints, right? I have lunch not because I'm hungry, but it's the time that fits between my two classes, right? I get up in the morning because of whatever other responsibilities and thing I have to do. Well, way back in ancient times, and by ancient times, I mean the 1970s, it was a completely different world, right? And one of the ways that it was different is when you graduated from high school, basically you had two choices. You either joined the Army or you went to a 4-year university. And when you went to a 4-year university, as a freshman at that university, you were required to take a psychology class. And really, that psychology class was mostly an excuse for professors there on campus to torture you, use you for experimentation. Probably one of the classic examples happened locally just at Stanford, the Stanford prison dilemma, right? Where they made some of them prisoners and some of them guards, and it got way out of control, and their injuries and all sorts of other crazy things that happened as a result of it. Most of you are familiar, or if not, I'll look it up. It's very, very interesting. But they did lots of other things too. One of the experiments they did is they took a bunch of freshmen and they locked each of them into a basement room. That room had no windows, it had no clocks, it had no television, it had a light switch, it had a refrigerator, it had books. And basically you, locked in that room, had no way of keeping time, and you lived in there for several weeks. Interesting, what they found is with those social cues removed, people actually got on a fairly consistent cycle, right? Again, if you think about it, this makes sense. Birds don't have alarm clocks, but they know when to get up in the morning. They know when to go to bed. They know when to fly south for the winter. When you remove the social constraints, people actually get on a fairly consistent schedule. Now, I don't mean that every single person on the floor all woke up at the same time. That's not what I'm talking about. What I mean is that you as an individual, you may get up at 7:30 every morning. Even though you don't know what time it is, you always would consistently get up at 7:30 in the morning. You would always eat breakfast at 9. You would always eat lunch at 2. You would always go to bed at 8:30, right, or at night, or whatever it is. People would get on a fairly regular schedule. And as was mentioned, what they found is one of the primary factors that influences this is the hormone melatonin. During the course of the day, we build up melatonin within our system, and that makes us fatigued. And when we sleep, that melatonin is broken down. Of course, as we've talked about, once we understand how the body works, we try to manipulate it to our advantage. So if you're having trouble sleeping, what can you do? That's one of the things you can do if you're having trouble sleeping. It is a horrible thing to do, but you can take a melatonin pill. Excellent. Melatonin makes me sleepy. So if I take a big pill of melatonin, it will make me sleepy and I'll be able to sleep. But here's the problem. Your body controls your hormones by a negative feedback process. So if you take a pill that massively increases your melatonin, yeah, you're going to be sleepy tonight. But what happens is your body goes, holy crap, there's a ton of melatonin in my blood. I need to stop making more. And it suppresses your melatonin production. So the next night, when you get ready to go to sleep, how much melatonin has your brain produced? None. So how tired are you? Not at all. So what do you have to do? Take another pill. And again, it produces this vicious cycle where you start suppressing your ability to produce your own melatonin, and you disrupt your ability to be able to wake. Hopefully none of you are taking melatonin, but one of the times when people will sometimes take it is when you travel to another country. Back in ancient times, you used to be able to fly on planes. And so with the weekend coming up, you might fly to England for the weekend. You get to England, you get out, you're ready to have dinner and party. The problem is everybody there is getting ready to get up and have breakfast and go to work. What do we call that condition? Jet lag, absolutely. Jet lag, right? Your cycle is out of sync with their cycles. So what some people will do is they'll take a melatonin pill the night before to try to reset their cycle. But remember the other thing that I said about the pineal gland. It is what some people refer to as your third eye. It is your first chakra. The reason I call it your third eye is because there are actually direct neural inputs from your eyeball to your pineal gland. Now, you obviously can't see, the shape of a blue boat by using just your pineal gland. So if your eyes were ripped out, you still wouldn't be able to see because you have a pineal gland. But it does receive light, and that light influences the production of melatonin. Right? It's one of the reasons why it's so important to not stare at your computer or your TV or your phone right before going to bed, because that light stimulates both your reticular formation, we'll talk about, but it also disrupts melatonin production in your pineal gland. But if you fly to England for the weekend, one of the things that you can do to dramatically reduce your jet lag is on your first morning there, get up at sunrise, sit out on the porch, and take in about an hour of sunlight. And as you take in about an hour of sunlight, that will help to reset your melatonin cycle, and it will dramatically decrease your jet lag symptoms. All right. Questions on that? All right, excellent. Just that simply then, we have finished off 2 of our cephalons. I think that was everything on our diencephalon. Have I missed anything? I got that all. All right, we finished our telencephalon, we finished our diencephalon. So that means tomorrow, Thursday, we will whip out our last 3 cephalons. We'll talk about our brain protections and our cerebrospinal fluid production and circulation, and then we'll finally get a chance to talk about our spinal cord. All right, any questions on any of that? All right, excellent. That is what I have for you today. Let me go ahead and clear this picture so we can see those structures more nicely. Let's go back to the illustration real quickly too. So here, this one. So again, notice, and I'll put circles. Here is our, actually, let's just do the highlighter. So anterior commissure, posterior commissure, pineal gland. So there we see those as well. Remember, we said the fornix points right at the anterior commissure, right? Corpus callosum points right at it as well, but it also points right at the pineal gland. And then from there, we get to the posterior commissure. All right. Okay, so questions on any of that? All right, so basically we have done everything from here up. We just have to talk about the rest of the brain stem. And like I said, these spaces that are filled with cerebrospinal fluid. So we're almost done with our anatomy and we're almost caught up. Like I said, we will start with anatomy on Thursday and then we'll move into the lecture on the spinal cord. All right, questions on any of that? All right. The way I figure it, you have 4 days off before all of your exams. This Wednesday, this Saturday, this Sunday, next Wednesday. 4 days where you're not being presented with new information before you have your Thursday lab and lecture exam and your Friday final. So make sure you're taking advantage of this time to master this material. Start looking ahead, making sure you understand these things. Remember, on Thursday, we're also going to try to get to the cranial nerves, see how far we get on that, although we may have to push that till Friday to make sure we finish the spinal cord first. We'll see how that goes, and we'll work our way from there. All right? Questions on any of that? All right, excellent. Then have a good day. Make good use of your day off tomorrow, and I will see you Thursday. Be happy, be healthy, be safe.